Good morning, everyone. Welcome back again. Now, from what I see, this is the last place that we had stopped. So, culture media is our next topic. So, guys, let's begin. First of all, yesterday I gave you an idea that culture media are nothing but the food for the growth of the bacteria. Okay, culture media are nothing but the food for the growth of bacteria. All right. Now, the next point is this. If you talk about the culture media, which are the food, just like humans have different requirements in their food. Okay. Just like humans like a different consistency of their food, like sometimes we like it mushy, sometimes we like it solid. So just like that, even they want their food to be different. So now technically any living being definitely requires proteins for their survival. So for that reason, what is definitely present in any culture media is peptone. What is nothing but proteins. Okay, peptones are the proteins. Is that clear enough, all of you? Now, once you know this, what is the next thing that you need to understand? There's something by the name of agar. Okay, something by the name of agar. Agar is not having any nutritional property, but it's simply to solidify the culture media. Just like our food, we want it solid or less liquid, something like this, right? So for that reason, agar is added into the culture media. Now, think logically. When do you think you will be able to see the motility of organisms? When the culture media is too tight or when the culture media is more liquidy? Motility, they have to swim, they have to show their motility. Obviously, reasonable answer is when the culture media is more liquidy, then I can see the motility. If you make it too solid, the organism cannot move. We won't be able to see the motility of organism. So as exactly as that, quite simple. The more the agar you add, the more the solid the media will become. So the least percentage of agar is 0.5%, which is semi-solid agar, and this can be used to visualize the motility. Sometimes if you expect an organism where you want to see the motility, then we can do this. 6% agar, it is to inhibit the swarming. It's also a motility. So more or less, let's just say it's to inhibit motility. Okay? So if you want to see motility, keep it liquid. If you don't want to see motility, keep it solid. But most commonly used, it is 2% agar. Okay, so most commonly used is 2% agar. That is there. I know this is not going to help you tomorrow in your life unless you come into the microfield. But again, if a question comes on this, you should have an idea how to answer. It's not very difficult, right? 0.5 means less solid, so I can see motility. 6% means very solid, I can't see motility. So that's about that. Now coming to the types of culture media, what do you need to understand? Culture media, first of all, called as simple culture media, they only allow the growth of simple bacteria. Okay, simple culture media allow the growth of only simple bacteria. Okay, so if I tell you in a food language, this is nothing but dal chawal. Okay, this is nothing but dal and rice, just very basic. Okay, people will eat it, but they will appreciate if you give something extra along with the simple media. Are you understanding? Along with the dal chawal, if I give you something like some crispy thing like papad, or if I give you some kind of a fry, if you're a non-vegetarian, some fry. Okay, something like this. If I give you along with that, then a lot of people will like that. See, exactly like this. If you're just giving dal chawal, then not a lot of bacteria will like it. So simple bacteria only can grow on this. Okay. Now, enriched media. Enriched, as the name says, it is enriched with something. Enriched media is enriched with something. So this enriched media, doctors, what you need to know is there is extra nutrients we add apart from the basic media. I just told you now, simple media or basic media, that is there. But along with that, there is something extra. That's called as, enriched media. As I told you, apart from dal chawal, you're having some pickles, you're having some papad, you're having some fries. All this will be considered as what? Okay, enriched media. Now, what is the papad, fries, etc. for the bacteria? Okay, blood. Okay, blood is required for them. If you give them blood, they'll be very happy. So many nutrients are present in the blood. Next, serum. You give them serum, they become super happy. You give them egg, right? They become super happy. A lot of people, in fact, eat omelette, etc., along with dal chawal. So they also want egg. So basically, this is what it is. Okay. Now, just for your reference in advance, I'll show you this. Have a look at this picture also. This picture is actually having meat pieces. It's legit meat pieces. That's why it's called as Robertson's cooked meat broth. Are you getting what I'm saying? It's called as what? Robertson's cooked meat broth. All right. So getting you back to this, whenever I say something is present in a culture media, it's genuinely present. When I say blood, it is blood. When I say egg, it is egg. So it's like that. So now the point is this: blood. What is the agar called as? As simple as that. It's called as blood agar. Nothing fancy in here. Lysed blood. Lysed blood. It is nothing but chocolate agar. Okay. When you add serum, it's called as Loeffler's serum slope. Okay, Loeffler's serum slope, in short called as LSS. We'll be studying this for diphtheria today. Loeffler's serum slope. Okay, thing like that you have just seen. I think now you can clearly appreciate why one is called as blood agar, why one is called as chocolate agar. Right? See guys, the blood agar is called blood agar because it's the color of blood. Chocolate is also blood, just the hemolyzed blood. The only difference being this is more chocolate in color. That's why they call it as chocolate agar, brownish. Next, selective media is the one. Okay, selective media. Now there are two, enrichment media and selective media. These are usually for some selected type of organisms, but enrichment media is a liquid, whereas selective media is the solid media. So solid media is for the growth of some specific organisms. So selective media is more commonly used. So please remember, selective is solid. Selective is solid media. Okay. Example, you're looking at LJ media right now. LJ media is the most famous one as such. Most of you may have studied this. It's for TB. Okay. LJ media, it is for TB, tuberculosis. All right. Next, if you talk about this, PTA stands for potassium tellurite agar. It's for the diphtheria. Okay, it is for the growth of diphtheria. Like that, there are these agars. I'm not going to read each name right now. Please understand, once we come to the respective bacteria as such in that topic, then we will, throw some light on this. So all of these are few selective agars that I have put in your notes that you need to definitely memorize this. Okay, so each bacteria has its own. I have cut it short to very few that are important that you should keep in mind. Okay, now how do anaerobes grow? I showed you this picture 2 minutes back. Anaerobes grow on this media, what we call as RCM. RCM stands for Robertson's cooked meat broth. Okay, RCM stands for Robertson's cooked meat broth. Now what I would like all of you to do is this. Okay, write it down exactly like this, how I'm showing you. P for Pikes media. These are transport media. So what is meant by transport media? Let's just have a look first of all. Transport media is only used to keep the sample alive for a longer time. Keeping it alive means what? Viable. Okay, so sometimes when we're collecting too many samples or whatever it is, right? The reason is if we have to save that sample for a while, then we use something called as transport media. So P for Pikes media, P for Pyogenes. Okay, same way, Vibrio cholera, V for Venkatrama Ramakrishnan, and C for Kerry Blair media. See, what are the two letters of Vibrio cholera? V and C. So you can use Venkatrama Ramakrishnan, another Indian contribution to this field, and Kerry Blair media. So these are the important ones that I want you to know. Okay. Next, Sachs media is for the Shigella. Okay, Sachs media, it is for the Shigella. So all I'm saying right now is this: Pikes media, Pyogenes. VC is Vibrio cholera, that is Venkatrama Ramakrishnan, and then Kerry Blair. And lastly, you have Sachs media, that is for the Shigella. These are the more important ones that they usually ask in exam, and you can very easily remember if you have read this hint I gave in the class once or twice. If you read it, you will get it in your head definitely during the day of exam. All it needs is you have to give it a reading, then eventually things will be piling up in your subconscious, and on the day of exam your subconscious is the one that will help you a lot. Okay, subconscious is the one that processes the info within few seconds and give you the answer. The thought that you get, right, that this must be the correct, it's your subconscious telling you that. And a lot of time we don't trust that guy, and that's a very big mistake. Subconscious processes things from what you have learned during all this session and gives you the answer faster. Because it's giving you the answer fast, we judge our own selves, thinking, oh, this may not be the answer. Let me think. When you start your thought process, a lot of time we mess up the right answer. So that's why I'm sure you have heard it from all faculties. You should always choose the answer that came first to your head and not change the answer. Okay? Not changing answers is one of the most important things. If I can assure this, if this point was followed by all students now, the pass percentage of this exam can be at least 5 to 10% higher. A lot of people who fall into 140 plus category, a lot of times they're not there because they haven't studied. They're there because they made mistakes, silly mistakes. Silly mistakes is what brings them there. Okay, tie your hands. If you can carry a rope to your exam, right, tie your hands after you have clicked everything. You have 10 minutes remaining, sleep. Do not recheck your answers, because in rechecking answers, if you change 20 answers, 2 will be correct, 18 will be wrong. And doctors anyway are not good in mathematics. You will choose the number 2. Okay, you'll be like 2 are correct, 18 are wrong, doesn't matter. Okay, you understand what I'm saying? Please do not rechange answers. All right, so anyway, back to our topic. Transport media, as I was telling you, they maintain viability, and then what we have mentioned is this is the way you can easily remember each of these guys. Okay, now this dude is coming up later. It's a chapter as such, very important, Enterobacteriaceae. So I'm going to cover this culture media in detail over there. Okay, we're going to learn about this coming up definitely later in the coming chapters. Now that finishes culture media. It's not a very difficult chapter. I hope you guys got that idea. Now here we are, bacterial genetics and antibiotic susceptibility testing. Okay, so what is this chapter as such? What you need to understand is this. See, number one, doctors, first point that I really want all of you to understand, it is this part, that is, when we make an antibiotic, two things can happen. Okay, it may kill the bacteria, so we call that as sensitive. That means the antibiotic is working, the bacteria are sensitive to our antibiotic, meaning they are responding, so they got killed. Okay, and antibiotics, some bacteria, they survive. Okay, and when the bacteria survive, we call it as resistant. Okay, so this is the two words that I want you to be familiar with: resistant and sensitive. Okay, resistant and sensitive. Are you all understanding what I just said there? Resistant and sensitive. Okay, so that's about it. This is the two things that are happening. Now, when you give an antibiotic, assume you gave an antibiotic for some organism and that bacteria somehow survived. Okay, so this is the bacteria that actually, saw the antibiotic. It is the one that is actually the one who has experienced our antibiotic and it survived somehow. Don't you think this guy is going to go and brag about his legendary stories, how he has survived this antibiotic? Of course he will, right? So for the betterment of their community, what will this bacteria do is, this bacteria goes and shares this knowledge. Okay, it shares the knowledge how it survived the antibiotic. So now the other bacteria who have not even seen the antibiotic will already become resistant to your antibiotic. Okay, I hope you guys got the idea what I said here. The other antibiotics, without even getting exposed to this, they are becoming resistant. So this chapter is all about this. How are these bacteria sharing the knowledge? What are the ways that they can share the knowledge? Is that understood? So that's what's the idea in this chapter right here. Okay, now that you may have gotten this idea, I would like to take this opportunity to explain to you a couple of things. Number one, please all of you draw this picture what I'm showing you on the screen right now. Just draw a circle. Okay, then if you have another color pen, first of all create 6 rings. Okay, 6 numbers, and then around three of them you should put some color if you can with another color. What are we learning here? I'll explain to you once you're done writing the stuff related to this. Okay, now that you must have done this, please write down these words what I'm showing you. Zone of inhibition. Zone of inhibition, wherever you have put some different color and that circle, right, just point to that and write that's called as zone of inhibition. And then the culture media, write down the word MHA, Muller-Hinton agar. MHA stands for Muller-Hinton agar. MHA simply stands for Muller-Hinton agar. So if you guys are done writing this much, now pay attention to what I'm saying next. What is this thing trying to teach us? Let's try to understand. See, first of all, what you have to imagine is, let's say I'm experimenting on some organism. Example right now, in my experiment, it is E. coli. You should assume I have already grown E. coli on this culture media. Okay, you have to assume I've already grown the E. coli on this culture media right here. Okay. Now the point is, what we are doing is all those 6 things that I've put there, they are the antibiotics. They're different types of antibiotics. So I'm checking in the laboratory which antibiotic can actually work against E. coli, which will not work. Okay. For that idea only, what we are doing is we are first growing the organism I want to experiment on, and then I'm putting 6 different antibiotics into the culture media. See, these antibiotics are not the oral form, not the IV form, not the IM or any other route, right? Because it's not a human being. It's a complete different route. It's a complete different route that we have to actually take here. So if you understood what I said just now, the point is these antibiotics are in a very different form for this experiment. They're called as antibiotic disc. Disc means something round, yes or no? So they are what? They are antibiotic disc. Now the question comes, after putting this antibiotics into the culture media, if something is happening around that antibiotic, do you think that antibiotic is doing something? Is it working or it's not working? You don't need to type anything, answer it for yourself, right? So I'm asking you one thing, that is, after adding antibiotics in the place where bacteria are already there, if something is happening, do you think that antibiotic is working or not working? The answer is it is working. Because it's working, something is happening in that area, yes or no? If it's not working, then nothing will happen in that area. So as simple as that, if you look into this picture, you can figure out 3 antibiotics are working against E. coli. Which? Number 1, number 4, number 5. Yes or no? Which antibiotics are working here? Number 1, number 4, and number 5. Why do I say they are working? Because they are killing bacteria around them. And that area where bacteria are getting inhibited or they're dying, it's called as zone of inhibition. That's what you pointed an arrow earlier and you wrote this is called as what? Zone of inhibition. Is that understood by all of you? Okay, this is known as the zone of inhibition. So now just below the picture, I would want you to make a note of this. 1, 4, and 5 are sensitive. 2, 3, and 6. 2, 3, and 6 which are not working, you should write the word resistant. Please do that. And if I ask you which is the best drug do you think that is working against E. coli, what should be your answer? Number 4. Why? Because number 4 is killing a lot of bacteria. Okay, so that's all about it. Please make sure that you put down those numbers. 1, 4, and 5, they are sensitive antibiotics. 2, 3, and 6, they are resistant antibiotics. Now once this is done, now is the important part. How exactly are these bacteria, as such, becoming resistant? How are they sharing information, what I told you about? This is what we are going to learn here, doctors. Okay, so let's start from here. First of all, assume that there was one bacteria here who had the knowledge about escaping antibiotics. Okay, let's just assume this was the bacteria, that circles are the knowledge. So technically this was the bacteria who had knowledge about many things. It's not always resistance. It can be, to make their own toxins more powerful so that they can affect the humans more easily. Okay, so that's that. Basically what we are saying is this, that is plasmids, etc. Or simply let's say that is the knowledge that they're possessing to escape antibiotics or to affect humans and cause a serious disease. All that information is present in the bacteria which I just have drawn for you, bacteria number 1. Now when this bacteria dies, all right, what happens is this information is released here as such in the environment. Okay, this bacteria has released this information in the environment. The bacteria number 2 comes near it now and it basically takes this information for free. It has taken this information for free. And when this happens, when the information is taken for free, it is called as transformation. Okay, this is known as what? Transformation. All right, these words are anyway written in your picture. This is nothing but transformation. Okay, now assume there is bacteria number 1, then there is bacteria number 2 as usual. Now the info is coming from bacteria number 1 into the bacteria number 2. Who is this green dude in between which is carrying that information? See, this green guy has previously affected this bacteria. So it's coming from this bacteria as such. The green guy has first affected the number 1. Now it is coming into the number 2. This bacteria, this dude is called as bacteriophage. Bacteriophage is a virus. Bacteriophage is a virus that you need to understand. Okay, so this is the virus that basically brings information from one bacteria to another. Viruses can even affect bacteria. Are you noticing that point? Don't get lost. Look at the big picture. Bacteria are getting affected by viruses. Viruses pretty much can affect anybody. They can affect human, they can affect animal, birds, bacteria, anything, right? That's your virus involved. Virus is called bacteriophages in what? The answer is transduction. What was transformation? One dude died, somebody picked up the knowledge, that's called as transformation. Now exchange is happening due to the virus. Virus has affected number 1. From the number 1 it's going to the number 2. That's called as transduction. This is with the bacteriophage. One word. From the environment, transformation. From the virus, transduction. And last and the final, doctor, is conjugation. Conjugation is happening between male and the female bacteria. Male and the female bacteria, and they exchange information by having something called between them known as pilus or conjugation tube. Sex pilus or conjugation tube. Basically the bacteria are mating. They're mating. Are you understanding? Male bacteria, female bacteria, they're mating, and here only the male can transfer the information to female because male has this genitalia. Male has this genitalia called as pilus. Are you understanding what I'm saying? After mating it looks like both of them have it, but actually only male has that genitalia. See, just like any animal, human anyway, males have genitalia that is coming out. It's just like that. Okay, sex pilus is just like that, and technically this is going to go into the females, right? Female bacteria. So here we go. We're just going to write all these things here one by one. First one, this blank, I already explained to you that it's happening between male and the female bacteria. What is this called as, doctors? This is known as the conjugation. Are you understanding what I said? Now, male bacteria is also called as F plus bacteria. Female bacteria is known as F minus bacteria. F minus bacteria. So there are 2 types of bacteria, male, female. Female is called as F minus, okay, and the male is called as F plus. You don't need to write it. Now here's where we will write something. Pay attention. There are 2 types of mating. Something very weird happens here. I just told you only males can transfer info. Yes or no? Only males, because they have appendage, they have a genitalia by which they can transfer it to others. Females cannot transfer this info. They can receive info, but they can't transfer it. So something very, very weird happens in bacteria. After the mating, the female, what is female? F minus now, right? So technically the female becomes a male. Are you understanding what is happening? So after the normal mating, what is happening? The female will become a male. Why is the female becoming a male? Because once it has learned something, now it wants to go and distribute this info, right? Now it wants to go and distribute this information. So the female has become a male. So that's about the normal mating. Is that understood, doctors? Now, next part is HFR conjugation. What is this HFR? High frequency. HFR stands for high frequency. This is the one in which the females remain female. They don't become males. Okay, they don't become male. So in the normal mating, female becomes a male, but in high frequency, female remains a female. Okay, so that's about that. That was conjugation. And I already have explained to you that if you're picking something from the surrounding media, it's called as transformation. What is transformation? Main word, guys, surrounding media, that is from the environment. Okay, that's your transformation. Next, coming to this thing, that is when a virus is involved, we call it as what? Transduction. So I want you to just draw this quickly, please. Bacteria 1, bacteria 2. Bacteria 1, bacteria 2. Who is transmitting this info? It is bacteriophage or a virus. Who's involved here? There is somebody that is bacteriophage or a virus. Okay, and when bacteriophage or the virus is transmitting this info, it is doing one of its life cycle called as lysogenic cycle. Okay, what is it doing, doctors? It's doing something by the name of lysogenic cycle. Lysogenic cycle. So that is that. It finishes this topic of resistances and everything. Now let's move on, guys. This is one chapter because I won't be able to help you. As simple as that. There are some discoveries of each of these guys. Okay, it simply is like reading. If I sit here, each guy contributed to something. There's no explanation in that, like why he contributed. We can't say, right? So he did something, he discovered something. So things are like that. Just for a reference, I'll give you one or two things that that you should understand. Louis Pasteur. This guy is known as father of microbiology. He's the one who proposed something called as germ theory of disease. For a long time, people didn't even know that something like germs or microbes exist. A lot of them thought, they thought that this diseases must be coming due to God or something like this. They had that kind of notions where they didn't believe there is something like this that can cause diseases. If somebody is infected, they thought some ghost came on them, right? Something like this. They had those kind of ideas. He was the first one to challenge all that and say, no, nothing of that sort is happening. It's basically because of somebody called as germs, all of this is happening. So he was the first one to actually say that something called microbiology is existing on Earth. Okay, because of him, let's just say that microbiology exists. And I'm sure you're getting a feeling that I wish he did not exist so that you didn't have this subject to study. Yes or no? So Louis Pasteur, the father of microbiology, please know he's the one that actually gave this germ theory of disease. He contributed to 3 vaccines called as CAR vaccines, cholera, anthrax, and the rabies vaccines. As a matter of fact, boiling of milk, what you call as pasteurization, has been named after him. Louis Pasteur. Louis Pasteur is pasteurization. Pasteurization. This has been named after him. This has been named after him. Are you understanding this? And he contributed to some other things also. Next, this guy Robert Koch is known as the father of modern microbiology. Then these are certain things that he has contributed to. But guys, as such in the recent exam, I haven't seen any question on the scientists and their contributions as such. Yes, it's not any disrespect or anything. We understand each of them has done something very, very important for humanity, right? Not just microbiology. These are all very big things for the entire humanity. So the point is this: questions are not asked because it's pure memorization point. Your exam is becoming reasonable now. They're asking the things which a doctor should be knowing. Okay, so that's that. Now coming to this last chapter as such in our general micro, it is called by the name of sterilization and disinfection. Sterilization and disinfection. Okay, now understand the point, that is when we want to kill somebody inside the body, when we want to kill organisms or infection inside the body, we can kill them by using antibiotics. But when we want to kill them outside the body, we have to use something called as disinfectants. Are you understanding this? So guys, what is the purpose? Let me give you one example so that you understand this. See, you must have definitely seen surgeons, right? Surgeons, whatever the equipments they're going to use in the surgery, they all need to be sterilized. Okay, why do they need to be sterilized? Because the person you're going to cut open, he's very prone to infection. Right now, skin is protecting you, nothing is happening. But when you cut open the body, are you not exposing the entire body to environment? So if that room is having organisms inside, they will penetrate, they will kill that dude by infection. Yes or no? So that's why we have to first sterilize that room itself. Operation theater also is sterilized. The equipments used by the surgeon are also sterilized. As a matter of fact, even the gown they wear is also sterilized. You must be all knowing now. Despite all of these things, infections can still happen. That's why we make sure that once a patient has been, has undergone a surgery, we shift them initially to ICU. Yes or no? In ICU, we are closely monitoring. We don't allow anybody to enter inside because it's more of a sterilized environment we try to maintain. Okay. Then once the patient recovers, we again send them to a ward after that. Yes or no? So all these things, please understand. In a daily life, this chapter is very, very important. But yes, we don't think about it because we are thinking about patients as such. But this is the indirect guy that is always there in the background, which is very important for us. Each of the things we are using a lot of times has to be sterilized. Without sterilization, you're actually causing infections in that guy, right? So that's the thing, sterilization and disinfection. So let's move ahead. What is the simplest thing that humans have learned that can kill microbes? The answer to that is heat. Okay, so this is where more questions can come from. Heat. Okay, now in the heat, when we talk about, there are 2 guys, one called as hot air oven, second called as autoclave. Okay, hot air oven and the autoclave. Now understand 1 or 2 things here. I want you to write this word. Hot air oven is called hot air oven because the temperature is very hot in this guy. Temperature is very hot in this guy. And what is autoclave then? Autoclave, in my language, if I tell you, it is nothing but pressure cooker. Okay, pressure cooker. Now I'll ask you one question. Let's say again, take the simple example. If you're making dal, right? If you're making sambar, whatever it may be, the point I'm asking is this: in which of the 2 vessels can you cook the curries or whatever it is faster? Is it in a normal vessel or is it in a pressure cooker? Obviously, the answer would be pressure cooker. That means the time taken for sterilization is less in the autoclave because it's a pressure cooker. Less time it will take, but this guy will take more time. It will take more time. Are you understanding? This is just the basic insight on what we are going to study in this topics: hot air oven and autoclave. Okay, other things, filtration and all, less likely to be asked in exam, guys. So whatever is higher yield, I will definitely ask you to study. Okay, now this chemical methods we are going to come across, one by one shortly. So keep marking whatever I'm telling you that you need to know. First of all, okay, they can ask you this. Make sure you know this. See, hot air oven. Hot air oven, as the name says, as I told you, it is very hot. Okay, it is very hot. Temperature, if you see here, it is very, very hot. How much is the temperature? 160 degrees. Guys, normally when we boil milk, etc., when you just see a little boil coming, we stop it. Yes or no? Maximum 20-25, half an hour, we will actually, keep it on the stuff. Point is, this time we are keeping things for how much time? 160 degree. That too for how much time? 2 hours. That too for the 2 hours. Are you understanding what I'm saying? So technically, this is the thing. 160 degrees for 2 hours. That's what is going to sterilize your substances. So first point that you need to understand is this, guys. Can you sterilize everything with this thing? The answer is no. Why not? The temperature is too hot and the duration is too long. Not everything can withstand that temperature. For example, let's apply our mind. Can I put plastic stuff into this? Answer is no. 160 degree for 2 hours, the plastic will melt. I can't, sterilize plastic materials by hot air oven. Rubber materials I can't sterilize by hot air oven. So two things at least that I can't use hot air oven for are plastics and the rubbers. Are you understanding this? But surgical instruments, which are usually metals, they can be happily sterilized by this. So most of the surgical things, okay, they can be actually sterilized by this thing. Is that understood what I said? Okay, so that's that. Now what is meant by control? Control means, guys, if you want to really check if your sterilization has actually happened, what do we do is, in that machine we throw some bacteria. After sterilization of 2 hours, we should check if those bacteria are still alive or no. If the bacteria are dead, that means your sterilization has happened properly. If those bacteria are alive, that means sterilization has not happened properly. So these are called as controls, doctors. Are you getting the idea what I'm saying here? These are called as the controls. So as simple as that, hot air oven is very hot. You can't sterilize some objects, as I told you. Okay, next, moving forward. Okay, so guys, when you have time, if you can, please give it all these things a reading. I'm teaching you the most important parts that are expected as such in examination. Okay, so here we are. Now we come down to this dude called as autoclave. So as such, we just covered what? We just covered hot air oven, and now I'm telling you autoclave. See, basically the heat is divided as such based on the temperature again. There are words what you're seeing over there now. So the point is this. Let's come to this part, autoclave. How much is the temperature in autoclave? It's not 160, it is 120 degrees, precisely 121. Okay, and it's for how much time? Only 15 minutes. Think of it, right? Usually in pressure cooker, when you make something, it takes only 15-20 minutes, right? So like that. And what is the difference in the pressure cooker? Pressure is added. And how much is the pressure? 15 psi. Okay, it is nothing but 15 psi. So that's all that you got to know. This is the most common temperature, and your exam will also ask this only. Okay. So understand, if you're increasing temperature and increasing pressure, the time needed will keep decreasing. Time will be decreasing. Are you understanding that? So anyway, this is the temperature you would definitely memorize. 121 degrees for 15 minutes at the pressure of 15 psi. Okay. What is the purpose of hot air oven? Understand it in words first. Uses are materials that cannot withstand hot air oven. Okay. Now the point is, what are the things? Example, non-sharp surgical instruments, plastic, rubber. These cannot withstand. These cannot withstand. Are you all understanding this? So that's about this, guys. Next, as usual, everywhere there would be somebody who's a control. Okay. Next, this is something that we have done now. This can be one MCQ that they can ask you. Please remember, who's called as cold sterilization. Okay. Cold sterilization. Cold sterilization is ionizing radiation. Radiation also kills microbes now. Radiation also kills microbes. So ionizing radiation, it is called as cold sterilization because it can actually damage or kill the bacteria without any increase in the temperature. It's not decrease, or it can kill the bacteria with decreased temperature. So the statements, either you remember that with decreased temperature, DNA damage with decreased temperature. This is called sterilization. What did I just teach you? Heat kills, organisms. But here it's not heat. It's not heat that is killing microbes. It's the radiation that's killing them. So without any temperature, we are able to kill it. That's why it's called as what? Cold sterilization. It's called by the name of cold sterilization. Okay. For example, again, rubber things like gloves. Gloves I can sterilize by cold sterilization. Are you understanding this kind of things? Okay. Now look at this picture and please write down. This chart is starting from here, weak to the strongest type of organisms. So technically the arrow is going this way, doctors, starting from here to here. Weakest to the strongest organisms in micro. Weakest to the strongest ones in the micro. Okay. So if you got that idea what I just said here, now pay attention. This is what I would want all of you to know. There are levels of everything in the world. Same way, there are levels of disinfectants also. High level disinfectant, this is the word what you need to know. Why is it called high level disinfectant? The answer is, it kills everything. It kills everything, including the spores. Spores are the most resistant in this chart what you're looking at. Okay, so who's going to kill it? It is high level disinfectants that kill it. And what is called as high level disinfectants? Plasma sterilization. Okay, there is something called as plasma sterilization. Okay, ETO stands for ethylene oxide, and aldehydes. Aldehyde means formaldehyde, etc. Okay, so you must have all done your dissections in the first year now. Why do you cry when you go near a dead body? Okay, I'm saying the cadavers, because what is present in that? Formaldehyde. Why is formaldehyde present in the cadaver? To prevent infection. If infection happens, the entire body would be decomposed. Yes or no? The entire body would be decomposed. It will not be available to do your dissections. So that's why we add formaldehyde to that. Are you understanding? To prevent any possible infection. So that's that, high level of disinfectants. Intermediate, why is intermediate called intermediate? Because this they may not kill. The last part, they may not kill, it's doubtful, but they definitely kill the other things. They're also quite strong. The weakest type of disinfectants are low level disinfectant, in which your alcohol comes, and alcohol is your sanitizer. Alcohol is your sanitizer. So what are the things killed by sanitizer? Only envelope viruses, bacteria, and fungi. It doesn't kill naked virus. I already told you back then in virology that your alcohol or sanitizer can only kill envelope virus, and we used it during corona because corona is an envelope virus. You can kill envelope. You can't kill the naked viruses by alcohol. They don't have that power. Okay, so anybody after this obviously will not be killed. Okay, so those are the different levels of disinfectants. Why? What was the purpose of classifying them like this? Okay, so I'll ask you one question. Let's say you have to sterilize your stethoscope. You have to sterilize your stethoscope because you're putting it on someone's bodies. ECG electrodes because you're putting it on somebody's bare body. Don't you think you should clean them? Forget about anything. The other person will not feel good because you're putting somebody's thing on their body, right? So obviously you have to sterilize these kind of things. Okay, but the point is, since these things goes just on top of the body, somebody's chest, etc., is there very high risk of infections? The answer is no. So you don't need to use like very high level of disinfectants as such. Okay, you don't need to use very high level of disinfectants. What are we going to use now? Low level disinfectant. For example, ECG electrodes, stethoscope, etc. What are we using? Please check it out. We're using what? Low level disinfectant. Okay, clear enough to everybody? Because the risk of infection is not there. All right. But if assume somebody has surgical implants, you're putting surgical implants into somebody, obviously this is dangerous. You're putting something inside the body, so you have to completely sterilize this. There should not be any risk of infection. So that kind of times we don't take any risk. What level of disinfectant we use? High level. That's all. So this thing is called as Spalding's classification. It's just teaching us in real life, when we are putting some things in the patients, etc., you should analyze what is this thing. Do you think there is a risk of infection? If I'm putting stethoscope like this on somebody, there's no risk of infection, even if I put it on another person. So technically, if you have to clean your stethoscope or whatever, simply use the low level of disinfectant. But if you're putting something inside the body, some implant, something like this, then you should definitely use high level, because that time there might be risk of infections. This is all called as what? Spalding's classification. Now, next thing. This is a straightforward thing, guys. There is no explanation, but do mark what I'm telling you, which are expected in questions. Starting from here, alcohol, you should definitely know. Ethyl alcohol 70% is used as sanitizer, right? So this is used as the sanitizer. All right. Next one, aldehyde. At least you should remember formaldehyde, as I'm telling you. Where is the question I've put? Formaldehyde. Formaldehyde is the one that is used in the fumigation of OT. Okay, you've heard the other claim of formaldehyde fumigation, and we should already know we all cried near the cadavers because of formaldehyde, as I told you, preserving the anatomical samples. Example, cadavers, right? So during the dissection, this is that give you that tears in your eyes. Okay, so things like this. And by the way, aldehydes, I already told you, they come under what level? High level. Aldehydes, they come under the high level of disinfectants. They're very, very strong. So formaldehyde, that's why you should definitely know them. Okay, now continuing the story from here, phenols. These are the intermediate level. What is phenol? See, maybe we didn't know these things, but now we know. One of them is this thing that is corrosive, so you can't put it on the skin. It's put on the floor. Lysol. Everybody have heard this now. Lysol. What is Lysol used as? For floor cleaner. Yes or no? Lysol is used as the floor cleaner. So that's your Lysol. Are you understanding? You can't put it on the skin as such. All right. See, this knowledge is very important, guys. Half knowledge, as they say, is dangerous. You must have definitely read in the news that people actually drank sanitizers thinking that it would prevent corona in them. You know this, right? People, sanitizers they have drank as such. Okay. They only help you outside the body. These are disinfectants. Yes or no? Next, the thing is antiseptics. They're not corrosive. That's why I can use it on my skin. Okay, so here we go. Savlon, Dettol comes under this. Okay, Savlon and Dettol, they're quite strong ones, right? Phenolic compounds, they're quite strong. So you have to eventually start memorizing this formula. All right, what are the formula for Savlon and this thing? Are you understanding? Next, coming to halogens, 1% sodium hypochlorite, super important. After formaldehyde, if I say this one is very, very important. See, this sometimes they may ask, sometimes they may not ask. That is there. But 1% sodium hypochlorite, definite. Okay, let's say HIV positive patient's blood fell down in the lab somewhere. Okay, obviously HIV is present in that blood, right? So when we have to clean that, we are going to use what? 1% sodium hypochlorite. Okay, next, betadine, I'm sure you know. Okay, betadine. So chlorine and iodine, they're mostly these two substances that are coming under the halogens. Okay, chlorine and then the iodine compounds. So technically in here, you should all be knowing iodine compounds. What is one iodine? Betadine, betadine, right? They should be very clearly known. This is nothing but a iodine compound as such. Okay, so zyada nahi hai, my friends. Let's look at it. How much did I tell you? Ethyl alcohol, everybody knows this is sanitizer. Formaldehyde, we all know. We have, as such, practically felt this, right? So this is anatomical samples. And F4F, as I told you, fumigation of OT. Formaldehyde, super important. 1% sodium hypochlorite, what we just learned, very important. Cleaning of the lab, right? Lab disinfectant. These are eventually you should memorize kind of things. Lysol, to okay. Nobody will even ask you, I think now, but you should know Lysol is a phenol, right? Lysol is a phenol. Dettol, phenol, right? So that's how they are. All words. So phenols. Okay. Next, halogens, what did I just tell you? Chlorine and iodine. Chlorine and iodine are the halogens, and in this you have to remember definitely sodium hypochlorite. See, there is a special smell that comes near swimming pools. Have you ever noticed? You know what is present in the swimming pool? Chlorine. Chlorine they add. Okay, so that's it. These are all the things that are there to prevent infections and everything. And 1% sodium hypochlorite you have to know. And these are the two things. Remember, in the high level, what did I tell you earlier? Plasma and ETO. Plasma sterilization and ETO. Right. So these are the two words that we just saw, and they're existing here that you need to sterilize. Learn eventually. ETO is what? Ethylene oxide, it's gas. So it's called as gaseous sterilization. Okay, gaseous sterilization. These are last minute things we have to see just 5-10 days before exam, guys. Don't bother memorizing right now. Please just write the word last minute review. Okay, these are the last minute revision things that you have to definitely see. Okay, high level. That's why I'm asking you to definitely study that. High level disinfectants are something you should be familiar with. Okay, so that's all about it. This is the chapter of sterilization. Clear enough, everybody? This is the chapter of sterilization. All right. Now, We will be beginning, with our next chapter, that is bacteria. So I'll give you a 2-minute breather, relax, then we will start. Okay. All right, guys. So let's go ahead. Now we are starting with our very important section, that is bacteriology. Definitely, it's the largest topic in microbiology, and of course, the score is also pretty much like that. Most questions come from bacteria. See, number one, we're going to go in a sequence. I hope you have already memorized who are the gram-positive bacteria. Yesterday I told you, so I'm just going to remind you again. First of all, what is the color of gram-positive? Purple. If you see purple, you should know it's gram-positive. Next thing is, what are the gram-positive organisms? Staph, strep, but they are cocci. How do you know? The name itself tells you that. Staphylococci, streptococci. Yes or no? So they are purple color organisms. Next, there are rod-shaped organisms, which were ABCD that I had asked you to memorize. Okay, so start from here. That is gram-positive cocci. Assume you have already done your gram stain, and this is the result that came. You know, if you see this result, gram-positive cocci, the organism is either staph or strep. Yes or no? The organism is either staph or strep. Is that understood what I said there? Okay, so staph would generally appear in clusters morphologically, and strep would exist in chains. Staph in clusters, strep in chains. So that is something that you should all be understanding. Okay, now how will I confirm whether it's staph or strep? Morphology is not confirmation. Okay, see, sometimes there can be somebody who doesn't know how to read that clearly. That means that guy can make a mistake. So morphology a lot of times may not be confirmatory. We have to do some other accessory tests. So what is the confirmatory test here that we need to do? The answer is catalase test. This is something from yesterday that we have learned. We are going to remind ourselves what's the function of catalase. It converts hydrogen peroxide to water, right? It converts hydrogen peroxide to water. So technically, if anybody is catalase positive, you will be seeing bubbles. You will be seeing bubbles. So is that understood? That's your catalase positive. You will be seeing bubbles right here. So what is something you need to know? Catalase positive organisms, they were memorized by that mnemonic called as places. Do you remember that word? Cat goes to places. This was the mnemonic I had given you back then. Cat goes to places. So what was in that cat goes to places? The organism called as staph. Yes or no? The mnemonic places. S was who? S was the guy what we call as staph. Are you understanding that? Right? So please write down. If you see bubbles, you should confirm it as staph. If you don't see bubbles, you should call it as strep. Okay, staph and the strep. Staph shows bubbles, whereas strep does not show bubbles. By this, we could easily figure out who is who. Are you all clear on this? What I just said? Staph shows bubbles, strep does not show bubbles. Assume for the continuity of this discussion. See, first of all, you can see the picture, catalase positive, catalase negative very clearly. Okay. Now the point is this, doctors. If we saw bubbles, we all know it is staph. Yes or no? If we saw bubbles, then we definitely know it is staph. So what is the idea? What is something that we all should understand very clearly is this. There are 3 species of staph that we have to learn. Aureus, Epidermidis, and Saprophyticus. I repeat, 3 species you have to learn anyway are what and what? Aureus, Epidermidis, and Saprophyticus. Okay, so these are the 3 agents that you got to know for sure. So technically, when a sample comes to me in the lab, see the beauty of gram stain. So many bacteria exist, but if I just got a purple color and round organisms, I have ruled out all this bacteria, and I'm just thinking between only 2 bacteria now, staph and strep. That's the beauty of this test, right? Purple color dikha and round shape, I know it is either staph or strep. I'm not thinking anybody else now. Okay, so it has given so much, simplicity, so much convenience to our testing. Yes or no? So once I know it's staph or strep, what is the test I perform? Catalase test. Catalase test told me it may be staph, it may be strep. Confirmatively. If you saw bubbles, it is staph. Next, I need to know the species of staph, which one it is. So I need to do the further testing. So look at the next test in the list, coagulase test. Okay, so what is the next test in the list? It is nothing but the coagulase test. Now, in the coagulase test, guys, you should understand, as the name says coagulase, you're going to look for coagulation. Some bacteria have enzyme called as coagulase, and what can they do, doctor? They can actually cause coagulation. They can actually cause the coagulation. So if you want to check if a bacteria can coagulate or not, what are you supposed to use in the testing? Blood or related products like plasma. Blood can clot, now water cannot clot. So what are you going to do? If you want to check if a bacteria has coagulase or not, take some plasma, put the organism into this. If it starts to produce clots or clumps, it means it has the enzyme coagulase. So who's coagulase positive, doctors? The answer is straightforward, staph or yes. Assume you did the test and bubbles were seen. You should know it is staph or yes. Nothing else needs to be known over there. Okay. Next is coagulase negative. Next one in the list is the coagulase negative. Negative means no clumps. This guy doesn't have coagulase. That's why there's no coagulation. No clumps. Okay. What is the word given if the result is coagulase negative? The organisms are called as cons. What is cons? Coagulase negative staphylococci. I said there are 3 species now. Aureus, Epidermidis, Saprophyticus. Okay. If you saw clumps, you know it is Aureus. End of the story. You're not going to do any more testing. That was the whole purpose of this test. Now, coagulase test, we're looking for coagulation. If you saw the coagulation, it is Aureus. If you didn't see coagulation, then that means it is either Epidermidis or Saprophyticus, together known by one single name called as cons. Who's cons? Epidermidis and Saprophyticus. Okay, is that understood by all of you? Now, how to differentiate the cons? You can see this test, write this test down. Staph epidermidis versus Saprophyticus, they are confirmed by antibiotic test. What is this antibiotics which will kill one of them, which will, make one of them survive? Novobiocin. We have to use this test called as Novobiocin test. So Staph epidermidis, Staph Saprophyticus, this is the story. Okay, Novobiocin test. So I hope you guys have written this. Let me just show you this through some pictures. Assume this is the guy suspected to be staph. You will do the test called as catalase test. And what is the result right now? Catalase positive. Next, what are you going to do? Coagulase test. What are you looking for? You're looking for coagulation. If you see coagulation like in this test tube right now, it is Aureus. Stop your testing right now because it's confirmed the patient sample is having what organism? Aureus. If you didn't see the clumps, that is known as cons. Who could it be? It could be epidermidis, it could be Saprophyticus. From this test, I will not be able to confirm because I know it's one of the two. I know it's one of the two, but I don't know which exactly. That's where we go and do this antibiotic test. By the way, do you remember a while ago when we were discussing the chapter, we have drawn this thing? I have told you we put antibiotics in the form of antibiotic disc. So there are two areas. I'm just showing you this. Can you guys answer for yourself only? Obviously, I won't be able to hear. Point is, which antibiotic is working or which one is sensitive? Working or sensitive is same thing. Na? The answer is number 2, because do you see around antibiotics, the bacteria are dying. The colonies of bacteria are disappearing. That means they are dying. So this antibiotic is working. Whatever it is, doesn't matter. Whichever antibiotic you put, that means this antibiotic must be working. Whereas number 1, it's not working as there is nothing happening around the bacteria. Yes or no? Around the number 1, nothing is happening. So that's that. Number 2, there are changes because it's sensitive. Now, if I tell you that this is the antibiotic called as novobiocin, if I tell you this is novobiocin, who could this organism be? Who could this organism be? Epidermidis, because you just wrote novobiocin sensitive. Who's the one who will die? Epidermidis. Who will survive? Saprophyticus. Okay, I hope you caught the point what I said. सबसे पहले basic सीखो. You should understand how to read this. If you're seeing number 1 kind of result, it means resistant. Nothing is happening with that antibiotic. You can see all bacteria same there. With number 2, you can see the changes. The bacteria dying in that area. That's why you see that zone of inhibition. Yes or no? So that's the basic idea in this topic right here. Everybody clear? Next, if you have gotten all these things, now the micro part over. Now is the clinical part. We'll come to the diseases of these organisms, starting with this guy, Cons Staph saprophyticus. Okay, so we are writing which guy right now? Staph saprophyticus. Okay, we are starting with Staph saprophyticus. Now, what is the thing that is important that you need to know about this guy? Guys, saprophyticus is the guy who will cause UTIs. What is the disease in one single word? It causes UTIs. It causes UTIs. Is that understood what I said? This is the organism to cause the UTI. Okay, this UTI has some buzzwords you need to know. Female, first of all. Female. And second thing is this. Apart from this, usually recently sexually active female. Recently sexually active females have higher risk of UTI from saprophyticus. It is called as honeymoon cystitis. Okay, this thing is called as what? It's called as honeymoon cystitis. Now, just for clarity, if both guys are given an option called as E. coli and saprophyticus, you should choose E. coli. Why? Because who's the most common cause of UTIs? Irrespective of what word is mentioned, it is E. coli. Most common organism for UTIs, it is which organism? E. coli. So even young, old, doesn't matter. Anything they say, E. coli is the answer. So if E. coli is not in the option, of course you choose saprophyticus happily. But if both are in the option, we better should choose what? E. coli. Clear enough to all of you? Next, what's the deal with Staph epidermidis? Okay, what's the story with this guy called as Staph epidermidis? See, epidermidis is going to cause endocarditis. Epidermidis is going to cause endocarditis. And you must all be knowing endocarditis is a disease that involves the valves of the heart. We all know now 4 valves exist in the heart. Left side you have mitral and aortic. Right side you have tricuspid and pulmonic. So these valves may be involved, and that disease is called as endocarditis. Now, I'll give you the clinical classification right away so that you guys are thorough and you understand and develop your thought process like that only. When a cardiologist comes to know that it's a case of endocarditis, what is the question this guy will be asking? Let's all first understand it right here. The first question is it the original valve of the person or is it the artificial valve we might have put in their body? So original valve is called as native valve. It's your original. Prosthetic, as the name says, prosthetic means artificial valve. Why are we learning it like this? Is because the causes, the organisms are different, and when organisms are different, the treatments may also be different. Are all of you understanding what I said there? The treatments may be different. So here's where you should first write this thing down. Okay, native is divided into 2 words. I'm just dictating that for you. Native is divided into 2 words: acute and subacute. Acute and subacute. So please write that down. Endocarditis, when we are writing, first word is what valve is it? Is it original or is it artificial? If it's original, it's called as native. In native, there are 2 headings: one called as acute, one called as the subacute. Acute and then the subacute. And in the acute, who's the organism? Staph aureus. Okay, in the acute, the organism is Staph aureus. The most common cause of acute is Staph aureus. Subacute, it is Strep viridans group. Subacute, it is Strep viridans group. So acute is over now. Acute is aureus, as I told you. Now I'm saying subacute. Subacute is which one? It's coming under native only. Strep viridans group. Strep viridans. Now coming into prosthetic. Prosthetic means artificial valve I have put into somebody's heart because their original valve is completely destroyed. Original valve is completely destroyed. That's why I'm putting an artificial valve. So write down the word early onset, late onset into that. Okay. Early onset, late onset word into this. Early onset, late onset. Early onset, it is epidermidis. Late onset is Strep viridans. When you learn this in internal medicine, they will tell you the definitions, what's called as early onset, late onset. Those things you will learn there. This is upfront. I'm giving you these things in advance so that you guys are familiar what is meant by this. So when valves are involved in the heart, you should know it is called as endocarditis. Now, first question, is it original valve? Is it artificial valve? If it's original, 2 words, acute, subacute, organisms are different. Prosthetic, you have these 2, epidermidis and viridans. Okay, so viridans is in 2 places, subacute also, late onset. In other words, it's in native valve also, it's in prosthetic valve also. Okay. Next, write down some statements, please. In SLE, there is one endocarditis called by the name of Liebman-Sachs endocarditis. Okay, there's something by the name of Liebman-Sachs endocarditis. Okay, Liebman-Sachs endocarditis. And if you have to choose one answer for endocarditis, who's one common agent in all of them? The answer is aureus. As such, the most common agent of endocarditis, it is your aureus. So I hope you guys finished writing all of this. So this is the actual clinical picture of when an endocarditis case comes. This is exactly how your thought process should be. Start with the first question. What is the valve? Is it original? Is it artificial? Then the respective words what I told you about, right? Details of this you will definitely learn in internal medicine later on. Now coming to Staph aureus. Now we are starting with the superstar bacteria called as Staph aureus. Now what do you need to know about this agent is this. Staph aureus is a normal flora in a couple of places in our body. One is in the nose also, and one is on the skin also. So it's a normal flora on our nose as well as in the skin. Okay, so that's your number one thing. Next, in its virulence factors, you need to remember something. It has something by the name of protein A. Okay, it has this thing by the name of protein A. And what is this protein A that you need to know? Protein A is a special thing present only in one strain of this aureus. It's called as COVAN1 strain. And if you remember our yesterday's class, I had told you this is an antibody. Upper part of antibody is called as Fab, and this is where antigen will bind. Antigen binding site is the upper part, yes or no? But here, the protein A allows this guy. See, what is the thing? Once the antibody binds to the antigen on the top, the cells will come and bind at the bottom, yes or no? The cells will come and bind at the bottom, and now they would have killed the antigen. They would have killed this antigen. But now, what is this guy doing is, it is basically, okay, binding to the FC. Protein A is allowing this guy to bind to the FC. So technically, nobody can bind at the bottom now, so it doesn't get killed. It is escaping our immune system by doing this. And thankfully, it is present only in one strain, not in all Staph aureus. Okay, so what is protein A doing? First of all, who is it present in? If they mention protein A for aureus, A for aureus. It binds to the FC fragment in an antibody. It binds to the FC fragment in an antibody. Now, next thing, start from here. Hemolysins are those factors. Hemolysis means RBC breakdown, right, doctors? So technically, hemolysins are those factors which allow this guy to break down the human RBCs or any RBC. Leukocidin, leukocytes means WBC. So leukocidin allows it to break down the WBCs. It allows it to break down the WBCs. And there's a name given to this toxin called as PV toxin. Okay, the name that is given to this, it's called by the name of what? PV toxin, which stands for Panton Valentine toxin. Panton Valentine toxin. So this was the guy who discovered this, so it's named after him. It's called by the name of what? Panton Valentine toxin. Okay, so I hope you guys got this idea, what I just said. Now moving on, check it out. Here's where you have to get an idea. The diseases that are caused by Staph aureus, they can be either due to the organisms or they can be due to the toxins. Any one of them can do that. See, other organisms may cause 1 disease, 2 diseases, 3 diseases like that maximum. This guy is having a paragraph of diseases. Some diseases are due to the physical presence of Staph aureus itself. Some diseases are due to the toxins it releases into our body. Are you understanding this? So some are toxin mediated diseases, some are organism mediated diseases. Both are different. Okay. Now, if you got that idea, check it out. What are the organism mediated diseases here? Okay, what are the organism mediated diseases? It's the most common cause of acute endocarditis, which we have already learned. So you better write the full word. It's the most common cause of acute endocarditis. Next, it's the most common cause of acute osteomyelitis. Okay, it's the most common cause of acute osteomyelitis. Next, lung infection is the most common cause of pneumatocyte. So technically, it's the most common organism of a lot of things. Because it's on the skin, it can cause 1,000 skin issues, right? So skin issues also, this guy's there. So it's causing a lot of things as you're noticing here. Okay. Next, toxin-mediated diseases. What is the next story here? Toxin-mediated diseases. So if you got this idea, what is this toxin-mediated diseases? Let's check it out. Okay, let's check it out. What do you need to know as such in the toxin-mediated diseases? First one is food poisoning. Okay, first one in the list is food poisoning, doctors. See, it is by one toxin called as enterotoxin. Okay, the name of the toxin is enterotoxin. Entero means GIT. That means this toxin is related to the GIT. And what is the disease with GIT? Food poisoning. Okay, so that's your food poisoning right here. Now, if you just remember that this guy is a superstar, I believe you can easily remember the points under this topic, right? The buzzword especially. First of all, it's a superstar. So do you think heating the food will kill it? Of course not. So what do you call such kind of things which are resistant? Heat stable. So technically, it's a superstar, right? It doesn't die like this. So heat stable. Next, it's the fastest onset food poisoning. It's the fastest onset food poisoning. How fast is it? Let's have a look. The onset of symptoms can be sometimes even less than 6 hours. You eat something, same day if you're getting a food poisoning, there are only 2 organisms to do this. One of them is aureus. So as a doctor, if you see some case of food poisoning, when you take a proper history, the guy says, no sir, today I had something outside, right? Every day I eat my food at home. Today I had outside and I'm having food poisoning. Same day if the patient came to you, it must be aureus or that somebody else I'm telling you about. Most other food poisonings come 1 day later. Okay, their symptom starts at least 1 day later, a lot of times. Okay, so that's about that. What is the primary feature in this? Diarrhea can also happen, but vomiting is the most important thing. Okay, it's seen with certain foods like potato salad, etc., etc. So technically, this is the 2 things you need to know: food poisoning, heat stable, and fastest in onset. Done. Next, let's go learn about toxic shock syndrome. Okay, toxic shock syndrome, toxin. So if you look at this word, shock is mentioned. That means this disease TSS. TSS stands for toxic shock syndrome. Toxic shock syndrome, and the toxin is called as TSST, which stands for toxic shock syndrome, toxin. Just highlight the buzzword what I said. That is shock. It leads to shock. And who are the people who are prone to getting this infection? The answer is females using vaginal tampons and not changing them in time. You must be all knowing, right? Vaginal tampons are things that are used during the menstrual cycle. So if the females don't change it in time, then it may get infected, and this may lead to even multi-organ failure, sometimes if you don't control, death also. So multi-organ failure, shock, etc., due to such a small mistake, right? Not changing the tampons on a regular basis. So that's your toxic shock syndrome. Next, another name in the list here, it's called as staphylococcal scalded skin syndrome. Okay, it's called by the name of staphylococcal scalded skin syndrome, sometimes popularly referred to as SSSS. Okay, so whatever it is, I want you to pick up the buzzwords, the important words first. If you read the name staphylococcal scalded skin syndrome, the word is scald. Yes or no? The word or the term here is what? Scald. What does scald mean? Scald means a burn, right? So technically here, the disease, it looks like as if the person got a burn. Okay, so this is your staphylococcal scalded skin syndrome. It's basically looking as if the person has gotten a burn. Clear enough to all of you? Okay. Next, what are some things you need to know here? What is the name of the toxin? It's called as exfoliative toxin. You must have learned in Dermana, the removal of skin is called as exfoliation. Removal of skin. So that's what's happening here. The skin is getting peeled off. That's called as staphylococcal scalded skin syndrome. Okay, everybody clear on this? Next, Let's learn the diagnosis aspect. Aureus is that organism whose name comes from aurum. Aurum is the name for gold. Okay, aurum is the name for gold. So if you see anything about this guy for its identification, it's going to be yellow. On nutrient agar, it produces golden yellow colonies. On blood agar, it breaks down RBC, releasing bilirubin, which leads to yellow color called as beta hemolysis. And then the selective media for this guy, it is called as MSA, mannitol salt agar. Either ways, anything you examine, it's always yellow. This is Aureus, yellow, golden yellow colonies. This is MSA, and this is Aureus, again yellow. All right, so whatever I'm saying, blood agar, it produces yellow. So that's the thing. Everything is yellow with Aureus. Is that understood what I said? Okay. Now, that being done, let's learn the next things in here. You must have all learned the drug that is used for the treatment of Staph aureus. It's a penicillin. If penicillin does not work, called as methicillin. If it does not work, then that strain is called as MRSA. Okay, that strain is called by the name of MRSA. What is MRSA? Methicillin resistant Staph aureus. Methicillin resistant Staph aureus. The drug of choice for which is vancomycin. Okay, drug of choice for this is vancomycin. See, why is the resistance happening? That means why is the Staph aureus not responding to our drug, which it was responding before? It has changed the structure. It has changed the structure. It has changed the site where methicillin comes and binds. It has modified the site where methicillin binds. So now methicillin cannot bind there. So what is it doing? They will ask you this question. What is MRSA due to? MRSA is due to mutation in something called as MECA gene. MECA gene mutation. This is your MRSA. And how exactly are you going to diagnose this MRSA? The answer is PCR for MECA gene. PCR can detect even genes. Okay. So PCR for MECA gene. This is the thing that you need to know as such. Clear enough to all of you? So that's about that. PCR. Done. Now you have something by the name of Versa. What is Versa? Vancomycin resistant Staph aureus. What are the drugs to be used for Versa? Two drugs, linezolid and daptomycin. Okay. If you have to choose one drug, drug of choice for MRSA is daptomycin. Sorry, Versa, it is daptomycin. And for pulmonary infections, drug of choice is linezolid. Okay, for pulmonary infections, the drug of choice is linezolid. For other infection, daptomycin, but for pulmonary infection, linezolid. Daptomycin does not work in pulmonary infection. That's why we have to choose another drug by the name of linezolid. So why are there so many resistances developing in this guy? Because they're exchanging information on a regular basis. Do you remember we learned these two words a while ago? Transduction and conjugation. Transduction and conjugation. Yes or no? So these are the two things that you need to be definitely thorough with. How does Staph aureus shares its info? The answer is transduction and conjugation. Okay, hope everybody is clear with this topic. All right. Chalo. Before we start our next topic, let's take our break. Okay. So we'll be starting by 11:05. All right. So we'll be starting again by 11:05. See you all then. Welcome back, everyone. So, continuing from the genus called as Streptococcus, let us see here what you need to know. Streptococci are these guys that you have to know that are catalase negative. Okay, these guys are catalase negative. I told you now you will not see bubbles with catalase negative organisms. So technically, let's assume we have done all that testing, gram positive result came, catalase test came negative. So now you know it is Streptococcus, but you don't know which Streptococcus. So what are we going to do? We're going to put that organism on something called as blood agar, right? That organism would be now put onto something what we call as blood agar. After you did not see bubbles, put it on blood agar. See, although there are three hemolysis, alpha, beta, and gamma. Gamma is not a hemolysis, it's a wrong name. There is no hemolysis as such that is happening. If there is no hemolysis happening, we call it as gamma hemolysis. Originally, there are only two hemolysis, alpha and the beta. Okay, and what do you need to know as such in this topic? Pay attention now. Guys, if you're seeing yellow color, that means it's called beta hemolysis, and it's due to release of somebody called as bilirubin. The color is turning yellow. So technically, these organisms are breaking down RBCs, because of which what is happening? Yellow color is getting released. That's called as what? That's called as beta hemolysis. Next, there's something called as alpha hemolysis. So what is the alpha? Alpha is the partial breakdown of RBC. Complete breakdown is beta. Partial breakdown is alpha. Alpha hemolysis is partial breakdown. And what you should remember is, guys, this will give you a greenish color. Greenish color is alpha hemolysis, and yellowish color is beta hemolysis. Okay. Now that you know, what is the idea here? See, why are we even doing all of this? First, once again, how did you confirm it's a Streptococcus? Catalase negative test. Done. No bubbles were seen on this test. Now you put the organism on blood agar. Either you will see yellow color or you will see green color. And actually, there are four organisms we can, kind of close down to if you actually get any of this hemolysis. So assume if you're getting green color, hemolysis, then that organism can be Streptococcus pneumoniae or Streptococcus viridans. That's at least how much I will come to know now, right? So I'll come to know pneumoniae or viridans. Next, if you're getting yellow color, there is somebody called as group A Streptococcus and group B Streptococcus, right? So if you see yellow, you should think only between group A and group B. If you see green, you should only think between, this guy that is pneumoniae or viridans. Is that clear enough to all of you? So that's about the idea. This is why we are doing this test. So let's go start writing these things down. On the blood agar, you get to see something called as hemolysis because of RBC breakdown. Beta hemolysis gives you yellow color because of what? Bilirubin. Bilirubin is released only after complete hemolysis. Okay, bilirubin is released after complete hemolysis. Now, next thing, what are the organisms in this? Okay, what are the organisms? Group A strep and group B strep. Both of them would be showing that yellow color. So by looking at the culture media, I will just know. See, if you see this yellow color, I will know it may be group A, it may be group B, but I don't know who it is exactly. Okay, so that's where I will have to do some more tests, and that is nothing but again the antibiotic test. Guys, this time what antibiotic we have to use? We have to use bacitracin antibiotic. See, earlier to differentiate epidermidis from saprophyticus, what did we use? We used something called as novobiosine. Here we have to use bacitracin. Bacitracin se pata chal jayega, we will come to know whether it's group A or group B. Group A strep is sensitive, group B strep is resistant. Okay, group A strep is sensitive, group B strep is resistant. Okay, next. Now the point comes down to this CAMP test. Okay, the thing is CAMP test. There's another test I can do instead of antibiotic test. I can do this test by the name of CAMP test. Now, what is the story with the CAMP test? Let's try to identify these doctors. In the CAMP test, group A strep is going to be negative and this guy is going to be positive. What is meant by this CAMP test? Look here, the picture is right below us. Like if you see the topic. Now, technically, if you see the shape of an arrow like this, if you see the shape of an arrow, that is called as what? That's called as positive test. That means it is group B strep. Positive reaction simply means arrow shape would be seen. Arrow is going to be seen. You better write it. What is meant by positive test? Arrow shape would be seen. If you don't see the arrow, then that means it is this guy, what we call as group A strep. Group A strep, no arrow. Group B strep, arrow. Okay. All right. So that's all about it. Now, continuing further, please understand and look here. Alpha hemolysis. Alpha hemolysis. It gives you greenish color. It gives you greenish color. And beta hemolysis gives you the yellow color. What were the two organisms with yellow color? Group A, Group B. Group A, Group B. And how were you differentiating them? Remember, the tests were either one antibiotic you can use or not an antibiotic test called as CAMP test. CAMP is not an antibiotic test, right? So non-antibiotic test. So in CAMP test, what were you looking for? Arrow. If you see arrow, it is group B. If you don't see arrow, it is group A. Now there's one more identification of group A. Group A is also called as pyogenes. So with the PY, you should remember a test called as PYR test also. It can show positivity. Group A strep can also be showing positivity for something called as PYR test. PYR test. Okay. Now next, continuing the story with the green colored roots. Why is the green color coming? Because of a pigment called as biliverdin. Biliverdin gives the green color. There are two organisms to show that green color: Strep pneumoniae and Strep viridans. Strep pneumoniae, or somebody called as Strep viridans. Is that clear to all of you? What I just said there. Now, what is the way I can differentiate these two? Same story. Use an antibiotic. Sir, I am saying if you see this green color organism, it can be pneumonia also, it can be viridans also. So just by looking at a culture media, I will not be able to say if it is this or that. So that's why what do you do? Once you know green color came, you already know it's either pneumonia or viridans. So that's why what you have to do now is put this guy on an antibiotic, right? Optochin antibiotic you'll be using this time. If the organism dies, that is Strep pneumonia. And if the organism survives, that is Strep viridans. Okay. So Strep viridans is resistant, Strep pneumonia is sensitive. Is that clear what I just said? Next, bile solubility test is another non-antibiotic test by which you can confirm. Same result. Strep pneumonia is sensitive in antibiotic test also. Strep pneumonia is sensitive in the non-antibiotic test also. Same result. And same way Strep viridans is resistant. Are all of you clear on this? Strep viridans would be resistant. All right. So Strep pneumonia is sensitive. Strep viridans is resistant. So that's all about this guy. What you need to know as such. Okay. So kya seekha humne? Let's all revise from the beginning. Okay. Assume a sample came to the lab. I have zero idea what it is, but my sample showed me this. It showed me this type of organisms. All of you now look here. It showed me this kind of organisms. What are you understanding when you see this purple and brown? Gram-positive cocci. So when you know it's gram-positive cocci, you already know in your head it is either staph or strep in the sample. Yes, you've already made your guess. So to confirm only, we did a test called as Catley's test. But if it's streptococci, no bubbles would have been seen because it is Catley's negative, no bubbles. So मुझे पता चल गया. Now I know that this is streptococcus I'm dealing with, but I don't know which species of streptococcus. So what am I going to do now? Put that guy on blood agar. Two things can happen. Either you will see yellow color, or you will see green color. If you see yellow color, you should know the organism could be group A streptococcus or group B streptococcus. If you don't see this, let's say you see the green color, then that means the organism is either Strep pneumoniae or Strep viridans. Because there are two organisms showing the same thing, how to differentiate them? We have to use some antibiotics. For the beta hemolysis, B for beta, B for bacitracin antibiotic is used. Whereas for the alpha hemolysis, you have to use optochin. Are you understanding? We have to use which antibiotic? We have to use somebody called as optochin antibiotic. So that's all about it. And you should know the results as I have told you eventually. Strep pneumoniae is sensitive whether it's an antibiotic test or a non-antibiotic test. Whereas beta hemolytics, bacitracin is the antibiotic. Second test is the CAMP test. What are you looking for in the CAMP test? Hemolysis in the shape of arrow. If you see this arrow-shaped hemolysis, you should confirm it is group B strep. If you don't see the arrow, it is group A strep. Clear? So this was just a revision of what I just explained to you with all these things. Now let's jump straight to this organism. Streptococcus pneumoniae, also called as pneumococcus or diplococcus. Okay, Strep pneumoniae, also called as pneumococcus or diplococcus. What do you need to know? Understand this. Unlike most of us think, okay, unlike most of us think that meningitis must be caused by Neisseria meningitidis, it's not. It's not the most common cause. Yes, Neisseria meningitidis also causes meningitis, but it's not the most common agent to cause meningitis. To your surprise, Strep pneumoniae is the most common cause of pneumonia also, and Strep pneumoniae is the most common cause of meningitis also. Are you understanding? So both fatal diseases are caused by the same agent. Strep pneumo is the most common cause of pneumonia also, meningitis also. Is that understood what I said? And if you remember yesterday's topic, I had told you that, you know, anybody who's causing these dangerous diseases, they are definitely going to have a capsule. They will definitely have a capsule. And what were the tests for capsules I asked you to memorize? You know, latex agglutination test or Quellung's test. Yes or no? Latex agglutination test or Quellung's test. These were the tests that I said. Modern test is latex agglutination test. Okay. And did I already teach you this? Yes or no? Capsulated organism risk increases if you remove the spleen. Splenectomy is always a risk factor for capsulated organisms. Okay. So that's that. I hope everybody got this much idea what I told you. Okay. Next, moving forward, what is another thing I want you to know? There's another infection caused by this, although there are others also, epiglottitis. Epiglottitis is also caused by this guy. Okay. Now what's the deal with epiglottitis? Epiglottitis, you should now know one thing. Earlier, H. influenza was the cause for epiglottitis. It was the most common organism. But now, Streptococcus has become not only Strep pneumoniae, the genus called as Streptococcus. This group has become the most common, organisms for causing what? Epiglottitis also. Epiglottitis also. Clear enough to all of you? See, I repeat this point last time. Epiglottitis earlier was caused by H. influenza more commonly, but now it is caused by Streptococcus. What is the reason behind this? H. influenza, we have already started in India. We are vaccinating the kids. We have started to vaccinate the kids against H. influenza. That's why. What is the purpose of vaccination? Why do you give vaccine? Vaccine is given so that the diseases don't happen, yes or no? So because of our vaccination drive, now H. influenza is not causing as much cases. Still, it can cause disease, but cases have subsided, and now slowly Streptococcus is rising. Okay, so this is something you need to know. And just for a reference, you will learn this in ENT also. What is one sign that you need to know with epiglottitis? There is something called by the name of thumb sign. Okay, there's something by the name of thumb sign. Thumb sign is seen in epiglottitis. All right, so that's about that. Next, as I told you, see, obviously vaccines are available. Those are the names of vaccines. See, check it out. On microscopy, if you look, this guy is called as diplococci. It is existing in pairs. Are you all able to notice? This guy is existing in pairs. Yes or no? So this guy is in pairs on microscopy. Hence, it's called as Diplococcus. It's called as Diplococcus. Now, if you talk about the gross appearance on culture media, it shows this colonies called as carom coin colonies. If you examine, it looks like what exactly? Carom coins. So it's called as what? Carom coin colonies. All right. So you need to recognize that this is your strep pneumonia. So carom coin colonies are a feature of Streptococcus pneumoniae. Okay. So hope you're done with this picture. As a reminder, once again, I'll go through this entire topic. Strep pneumoniae, also called as Diplococcus. You saw the picture. Why is it Diplococcus? Because it exists in pair, right? That's why. Of course, the name has pneumonia, so pneumonia to karega hi karega. It will cause pneumonia. But it's also the most common organism to cause which other disease also? Meningitis in all age groups except neonates. Neonates means somebody else will cause it. In the neonate, somebody else will cause the meningitis. There's a special term for it called as neonatal meningitis. All right, are you understanding what I said? Okay, let's move. Now coming to the term called as Streptococcus viridans. Streptococcus viridans. These are generally the normal commensals of our mouth. Okay, especially strep mutans. You're going to remember exactly like this. M for mutants, M for mouth problem. What is the mouth problem? Dental caries. M for mutants, M for mouth problem. Dental problems are usually due to Streptococcus viridans. It's a group, guys. It's a group. There are multiple guys that come under this group. Okay. Next is Streptococcus sanguis. S for sanguis. S for it is the cause for subacute bacterial endocarditis. See, the better word is Streptococcus viridans group, but if they give one organism, it can be sanguis. Subacute bacterial endocarditis. Okay. And what is the other word that you need to write here just below this? The same guy is also causing what? Late onset prosthetic valve endocarditis. So I'll remind you once again, just hear me out. What did we learn with endocarditis? First question to be asked, which valve is there in the patient? Is it native valve, or is this the other one called as prosthetic valve? I told you in prosthetic valve, after you put a valve, now the countdown begins. When is the endocarditis happening? After you put the artificial valve. Is it early or is it late? See, with the original valve, we're not counting time now. That is there in our body since our childhood, since we were born. When do you count the time the infection will come? After you put an artificial valve. And what is artificial valve? Prosthetic valve. Late onset prosthetic valve endocarditis. Okay, so I hope that's clear enough to all of you. Done. Let's move. Group A streptococci called as strep pyogenes now. Group A streptococci called by the name of pyogenes. What do you need to know about this? See, I have described all these words already. They're yellow, they're bacillus insensitive, and P for pyogenes, PYR positive. So I gave you all these words earlier also. Now understand, when I'm using the word like group A strep, group B strep, on what basis did they divide them into groups and this thing's what we call as the subtypes? Okay, so sometimes they may ask this, so that's where I want you to be ready even if they ask you this. See, first of all, the groups are called as Lancefield groups. Okay, the groups are simply called as what? They're called as Lancefield groups. Clear? How many are there? A to V. Forget I to J, I mean exception IJ, etc. A to V, group A, group B, group C, group D, group E, like that. So many groups are there, but we are not going to study so many. Just two you have to know. Group A, group B. Are you understanding this? The most important ones for us medically, group A and group B. These are the two I'm asking you to study. Okay. Now this Lancefield grouping has been based on the carbohydrate antigen. Okay, so imagine this is a organism. This is one streptococcus. This is one streptococcus. This is one streptococcus. So all of them are streptococci. They may have different types of carbohydrate antigens on them. For example, this is one carbohydrate antigen. This is one carbohydrate. This is one carbohydrate. Okay, all are carbohydrates only. The point is they are a bit different. So based on the carbohydrate antigen present on their surfaces, we have classified them into different groups like this. Okay. Next, there are types within each groups. Especially group A has more than 100 types. These are called as Griffith types. This is due to M protein. M protein. Okay, so you should be memorizing it exactly like I'm saying. London City. This is your first word. L for Lancefield group based on carbohydrate antigen. Okay. London City. Good morning. Okay. So this is the word we are going to write here. London City. Good morning. So what is good morning here? Griffith types and M proteins. Griffith types and the M proteins. So London City. Good morning. This is just a mnemonic for us to recall these things what I mentioned here. Okay. So now that you're done here, let's continue and understand something. First of all, if you talk about virulence factors. Okay, if you're talking about virulence factors, a very important virulence factor here is called by the name of streptolysin O, in short abbreviated as SO. Okay, in short it's abbreviated as the SO. Just the short form. The first letters SO means streptolysin O. Okay, now this guy is cardiotoxic. This guy is cardiotoxic. Please understand this thing. This guy is cardiotoxic. Okay, is that understood what I said? So cardiotoxic is streptolysin O. So assume this streptolysin O came in your body. Okay, don't you think your body will make antibodies against this streptolysin O? They will, right? They will make antibodies against the streptolysin O, and these antibodies are called as antibodies, or let's just say, let's take another color, antibodies against what? Streptolysin O. In short, they're called as what? Antibodies against the streptolysin. See, when will you form antibodies? Only if you're exposed. Only if you're exposed, you will form antibodies. Are you following this? Only if you're exposed, you will form what? You will form antibodies, as I'm telling you right now. Okay. So this is the idea. When do you check this? I'm sure you have definitely studied this, in your curriculum. ASO titers. ASO titers. ASO titers are used in the diagnosis of which disease? Rheumatic fever. They're used in the diagnosis of which disease? Rheumatic fever. And what is the organ involved in rheumatic fever? Heart. Are you understanding why we check for this antibodies? Because in rheumatic fever, heart is damaged. If heart is damaged, I want to know, is this the organism which is there in my body? Are you understanding all of this? So this is your ASO titers, what you study in this topic. And as a matter of fact, there is this criteria, all famous criteria called as Jones criteria. For the diagnosis, we have something by the name of what? Jones criteria. Is that clear enough to all of you? So that's the thing that you need to be thorough with. Okay. Next, let's move on. Write down one more thing here. Something by the name of streptokinase. Okay, streptokinase. Streptokinase is an enzyme as such. Streptokinase is an enzyme as such that is by this organism called as Strep genus. So technically, this enzyme has ability to break down clots. What is a clot made up of, doctors? Clot is made up of a substance called as fibrin. A clot is made up of a substance by the name of what? Fibrin. Are you understanding what I'm saying? So technically, the clot's breaker would be called as fibrinolysis, fibrinolytic or fibrinolysis word. Are you understanding this? Okay. So if you got it, what did I just say? Pay attention. Streptokinase is an enzyme that is used to break clots. We use it in medicine as such. You must have definitely studied when there is a heart attack called myocardial infarction in someone. We break the clots by using this thing. Okay, streptokinase. It's called as thrombolysis. The process is called as thrombolysis, or the drug is called as thrombolytic drug. So technically, it is from this guy. Okay. Next. Now, the next toxin that you have is streptococcal pyrogenic toxin. Streptococcal pyrogenic toxin. Pyrogen matlab fever, fever causing toxin. And guys, it's also called as erythrogenic. Erythro means red. It causes vasodilation. Because of the vasodilation, you get this very red colored tongue, described as what? Strawberry tongue. And it's a very classical feature of this disease called as scarlet fever. Okay, scarlet fever. So this is something that you need to know. Vasodilation is happening, which gives that very red tongue appearance called as strawberry tongue, which is the diagnostic feature of what? Scarlet fever. Okay. Now, you must have definitely studied this scarlet fever is one disease causing strawberry tongue, another is called as Kawasaki disease also. Yes or no? In medicine, you must have definitely read Kawasaki disease, another disease to present with this strawberry tongue. Alright, so that's all about it. So one fancy name fever you have learned. Scarlet fever, it is by pyogenes. Scarlet fever, it is by pyogenes. Now write down this thing. First of all, just fill up this blank. Pharyngitis or sore throat is a common infection that is caused by this guy. Strep pyogenes can cause pharyngitis or sore throat. Okay, pharyngitis or sore throat. Or it may cause some skin infections. Skin infections like impetigo, erysipelas, etc., etc. These are all the skin infections. So guys, once this happens, there are some complications that can happen following this, and the complications are, right in front of you: rheumatic fever and PSGN. Rheumatic fever and PSGN. So I already told you in rheumatic fever we check for antibody, ASO titer, right? ASO titers. Same like that, we can check for one more antibody titer called as anti-DNase titer for PSGN diagnosis. Okay, so if you have written that, let me just clarify some things to you. Sabse pehle, this is the point I would like all of you to understand. See here. When somebody gets infection with this, what are they getting? They're getting either sore throat or soft tissue infection, skin or soft tissue infection, abbreviated as SSTI. You already saw na? Skin or soft tissue infection. Isko hum short mein kya bolenge? SSTI. So patients will get sore throat or SSTI. This will get complicated not because of the organism, but because of excess body reactions, hypersensitivities. Let's say after this infections, if a patient get type 2 hypersensitivity, they will develop which disease? Rheumatic fever. After this infections, if they develop type 3 hypersensitivity, they will develop what? PSGN. What is PSGN? Post-streptococcal glomerulonephritis. Post-streptococcal glomerulonephritis. Are you understanding what I'm saying here? So now, first of all, remember, rheumatic fever is diagnosed by what criteria? Jones criteria. And another important thing in the criteria is ASO titer. ASO titer. Clear enough to all of you? So Jones criteria and then the ASO titer. Now, once you know this thing, that was heart disease. Next is the kidney problem, glomerulonephritis. Nephritis, itis wala word aa raha hai. It is a nephritic syndrome. PSGN comes under nephritic. So you must have all read something like nephrotic syndrome and nephritic syndrome, proteinuria and all those things. So PSGN comes under nephritic syndrome. And you all have to know one thing, dear doctors. Nephritic syndrome mein kya hota hai? There's always hematuria, RBC cast, etc. All those things what you study, right? Hematuria happens as a classical feature of nephritic syndrome. And now, what is the point to be noted? The urine color is so dark in this topic that you must have studied. There is this fancy word to describe it: cola colored urine or cocoa colored urine. Okay, cola or cocoa colored urine. Chocolate color, matlab cola dark color. Right, so this has come in question before. So cola or cocoa colored urine, this is what you got to know. Okay, so that's about it. Next, let's continue. We have finished the diseases about this guy. I've already given you this transport media. Today we learned it, remember? Pikes media. P for pikes, P for pyogenes. P for pikes, P for pyogenes. Done? Last organism or second last organism, that is Enterococcus in the topic of streptococci. Now guys, what do I want you to know as such in this topic? See, check it out. See, Enterococcus, this organism is known for mainly endocarditis, that you should know. Endocarditis. And endocarditis in which people specifically? IV drug abusers. IV drug abuser, in short we can call them as IVDA, IV drug abusers. So guys, endocarditis, if it involves the left-sided valves like mitral or aortic, most probably it can be Enterococcus. If right-sided valves are involved in the IVDA, then that is mostly Staph aureus. And if one option you have to choose, they didn't mention left, right, anything. They just mentioned IV drug abuser getting endocarditis. Overall, most common cause in IVDA it is aureus again. Okay, it is again going to be aureus. Even in drug abusers, the most common cause of endocarditis, it is Staph aureus. Okay, you guys understood what I said there? So this guy is going to be Staph aureus. Done. Next, sir, group B strep. What's the story with the group B strep that we need to understand? Let's all look. So doctors, when I say group B strep, A. galactiae. Group B strep is which one? Group B strep is A. galactiae. See, what you need to understand is one word as such. This guy is the most common cause of neonatal meningitis worldwide. See, let's recap something. Who's the most common cause of meningitis overall except for neonates? Strep pneumoniae. Remember, strep pneumoniae is the most common cause of meningitis in all age groups except for neonates. So, neonates mein kaun karega? Who's going to do it in the neonates? The answer is group B strep. Okay, group B strep. Now, if you're talking, in India, the cause is slightly different. See, in India, the cause is Klebsiella. The most common organism is Klebsiella, or there's one more that is E. coli. So Klebsiella and E. coli, these are the two that we have here. Klebsiella and then the E. coli. These two organisms are very important for the neonatal sepsis or neonatal meningitis, and both of them happen to belong to one family called as Enterobacteriaceae. So better answer, Enterobacteriaceae would be our most common, family as such over here. Okay, that's our Enterobacteriaceae. Baki, how to identify this? I have already given you the identifications. Remember, I have told you we can identify this by Bassett-Tracin test or the CAM test. Bassett-Tracin test or the CAM test. So yaad rakhna hai, everybody. Let's repeat. The most common cause of meningitis, any age except for neonates, it is strep pneumo. Okay. In neonates worldwide, it is group B strep called as A. galactiae. But in India, they are the second important cause. They are not the most common. Most common is which one? Enterobacteriaceae members like Klebsiella or E. coli. Klebsiella or the E. coli. So that's about it. That's the guy right here. Okay. So we will be studying our next topic, that is gram-positive bacilli. After a relaxation of 5 minutes. All right, so this 5 minutes, up to you guys. You relax or you just turn pages and see what did we cover as an overview. Okay, utilize the time. Okay, so let's begin, guys. Now, next, what we have here is Bacillus anthracis. We are starting with the next genus, that is gram-positive bacilli. So this is nothing but our, genus, that is gram-positive bacilli. So Bacillus anthracis, dear friends, what disease does it cause, etc. Of course, anthrax is the name of disease. Quite easy to derive from the name of the organism. So first, you write this point. This is a spore-forming organism. This is a spore-forming organism which is non-bulging spores. Okay, it is non-bulging spores. So is that what clear enough to all of you? It is non-bulging spores. Next, if you talk about the virulence factors, yesterday we learned that it has a capsule, and in fact we said that it is the only organism to have a protein capsule which was made out of something called as D-glutamate. Yeah, we have learned this in the capsule topic, that this is the one that is having that D-glutamate capsule. Okay, let's now come to the disease right away. If you talk about the disease, basically this guy, it is a zoonotic disease, meaning it comes from animals to human. Okay, so it comes under zoonotic kind of diseases which comes from animals to human. So for a rough idea, and that's actually better for us to know this to understand the disease better, who are the animals from which humans can acquire this? Answer is cattle. Okay, so this disease can come into humans through cattle. Is that clear enough? Now, talking about the disease called as anthrax, what are the types of anthrax? Let's learn. Cutaneous anthrax, they never ask that name, too easy. Rather, they will ask you one word, Hyde-Porter's disease. What is the word we are saying, doctors? Hyde-Porter's disease. Hyde-Porter's disease. Why is it called so? Because first of all, it's a skin form of anthrax. And who's going to get this? Somebody who's transporting skins of animals. What is skin also called as? Hide. Yes or no? So basically, skin is also called as hide. So if I'm somebody who's transporting skins of animals on my body, then I will get it. So the question comes, why would I be transporting skins? Because leather is made out of animal skin. Yes or no? So Hyde-Porter's disease, when I'm using that word, you all have to understand the point. What is that point we need to know? The point is this. It is the one that is a skin disease manifesting as a eschar. So please, you write this word and write what is this disease? Hyde-Porter's. Write down the full word, okay? It's called as Hyde-Porter's disease. Hyde-Porter's disease. Okay, that's clear enough to all of you. This is called by the name of Hyde-Porter's disease. It presents with this eschar. Okay, now, what is the next type of anthrax? The next type of anthrax is called as what? It is known as Wool-Sorter's disease. Okay, so this is the next disease by the name of Wool-Sorter's disease. See, I just told you, cattle means cow, sheep, etc. And the wool comes from the sheep, yes or no? So somebody who's sorting the wool, obviously to make jackets, whatever it is, the wool is used in so many things now to keep us warm. So the point is, Wool-Sorter disease, a farmer who's sorting the wool may inhale the spores of this organism. The farmer may inhale the spores of this organism. And when the farmer inhales the spores, what is that farmer going to get? Pneumonia. Okay, that farmer is going to get what? Pneumonia. Is that understood what I said? So Wool-Sorter's disease, basically it is coming by inhalation of spores, which is going to give you what? Pneumonia. And it's fatal, of course. Okay, this thing is fatal. And because of this nature only, this is used in bioterrorism. Okay, this is a potential agent that we can use in what? Bioterrorism. Clear? So this is all that you got to know, and it has happened in USA earlier. What some terrorist groups did is they sent envelopes containing spores of this organism. They sent envelopes containing spores of this organisms as such to random people, and they died because they inhaled the spores. Okay, so biowarfare, as you may be knowing, right? This is the future of warfare now. Okay, release infections which can kill people, and there's nothing better than pneumonia. See, if people have bad intentions, if pneumonia is spread to few people, they will spread 1,000 others by coughing, sneezing. Yes or no? What is COVID? COVID was nothing but a respiratory infection. Very easily it could spread. Are you understanding this? Same way, these kind of diseases can be used for this. So once again, a quick recap. This guy is coming through cattle. Okay, क्या क्या हो सकता है? If you're transporting the skins of those animals called as hide porter's disease, you will get this eschar I told you about, right? And if you're talking the fatal form of this disease, it is pulmonary anthrax, which is also called as what? Wool-Sorter's disease. It's called as the Wool-Sorter's disease. This comes by inhalation of spores. Okay. Chalo, next one, McFadden's reaction. Next one in the list is McFadden's reaction. So what's the deal with the McFadden's reaction? What do you need to know? I've already told you this story. McFadden's reaction is done for the protein capsule. Don't you guys remember that story? Quellung's reaction is for the sugar capsule LPS, whereas McFadden's is for the protein capsule. Now the appearances of this guy. Okay. On the gram stain. See, first of all, we are looking at a rod-shaped organism. So these are 4 different bacteria. I mean, 4 different Bacillus anthracis. But the way they have joined together, they have arranged themselves. It looks like a bamboo stick. Yes or no? It looks like what? It looks like a bamboo stick. So simply put, this is called as what appearance? Bamboo stick appearance. This is called as the bamboo stick appearance. Clear enough to all of you? On the gram stain, what do we get to see? Bamboo stick appearance. Now let's look at this in the actual image, how it looks like. See guys, this is the bamboo stick appearance what you're looking at. And if you see the pictures, inside them there are these holes kind of things. What are these things? These are the spores. These are the spores. And when you look at the spores, the idea is this, dear friends, they're not bulging out of the organism. They're inside the organism. Yes or no? They're not bulging out of the organism. Whereas if you see this side, what is the story this side if you notice? Okay, this is the rod-shaped organism. You can see a rod-shaped organism, rod-shaped organism, rod-shaped organism. But then the spores are bulging out of the organism. They're much bigger than the organism itself. They are called as bulging spores, which are feature of Clostridium. If you may have read these words like tennis racket spores, drumstick spores, that's Clostridium spores because they look like tennis racket or the drumstick. Drumstick what we use as such. Are you understanding what I'm saying? So this is the typical appearance of Clostridium, whereas Bacillus will have which kind of spores? Non-bulging. This is non-bulging spore, whereas this is a bulging spore. Very clearly, I think you can identify these things. Okay, so that's about that, doctors. Okay, next, coming to the penicillin agar. On the penicillin agar, this organism shows something by the name of string of pearl appearance. String of pearl appearance. So these appearances are quite important. Look at this thing. Another one what I'm showing you right now, this is the one, this is called as inverted Christmas tree appearance. Okay, this one is simply called as inverted Christmas tree appearance. I hope you can easily figure that out. This is described as the inverted Christmas tree appearance. Okay, that's about that. Next, moving on. So these are the important identifications. You can't forget bamboo stick or Christmas tree, etc. These are very, very high yield. You got to know for sure. Okay, now coming to this part, Bacillus cereus. Bacillus cereus. What do we need to know about this organism? One word. The moment you hear the word cereus, you have to know Chinese fast food poisoning. What do you need to know, doctors? Chinese fast food poisoning. So this is the thing that you need to know along with this guy. Okay, let's move on. Now coming to this, there are 2 types of presentations with this. One is a diarrhea, one is a vomiting. Okay, one is a diarrhea, another is a vomiting. So these are the 2 types of presentations you can expect with this. One being diarrhea, one being vomiting. Okay. Now when you talk about the emetic type first, emetic means vomiting. Now this time it's copying someone. Right? This Bacillus cereus, it's copying someone. And you should remember, if it wants to copy someone, who do you think is the best guy to be copied? The superstar. And who's the superstar, doctor? Staph aureus. Yes or no? The superstar is called as what? Staph aureus. So technically, it is copying Staph aureus. And you need to know these words. Heat stable toxin. Did we not study this? I told you when it's copying superstar, it's copying it thoroughly. And how was the superstar? Superstar does not die by heating the food. It was heat stable. Next, how was the onset? Fast. This guy is also fast. Remember when I was teaching you aureus, I said on the first day, if you see somebody coming to you with food poisoning, because that day only they ate something, that means who must be present in that? Aureus must be present in that, right? Aureus must be present in that. Are you understanding what I just said? Aureus must be present in there. So heat stable, that is the first word. And second, this is this. So who are the 2 fastest food poisonings? Answer is aureus and cereus. Originally aureus is aureus. This guy copies aureus. Aureus and cereus. Even the name it has kept is like that. I want to copy aureus. His name is cereus. Okay. So this is the guy. So this guy seriously copies others. That's how I remember. Cereus seriously copies others. So here we are, emetic type. Okay. ये आता कैसे? How exactly? How should you know if it's aureus or cereus as your answer? If everything is same, vomiting, vomiting, fast, fast, heat stable, heat stable. Then how should I answer aureus or cereus if both are in the option? Answer is reheated foods, Chinese fried rice. Okay, Chinese fast food. Reheated foods is the buzzword for this guy. And sometimes it may copy others, causing diarrhea. And when it is causing, this is less important. My opinion, emetic type is more important. Okay, diarrheal type, if you're talking, it's copying somebody else. This time, who is it copying? Somebody called as Clostridium perfringens. Okay, so this guy is copying which guy now? Clostridium perfringens. And basically, you need to know this comes much later. It is slow onset, and it is heat labile. So every word is opposite. If this is heat stable, that is heat labile, opposite. If this is fast, this is slow. Okay, if this is vomiting, this is diarrhea. So pretty much everything is opposite here, but this is less likely to be questioned as such. So it comes by consumption of improperly cooked meat. Okay, it comes by consumption of improperly cooked meats. So that's about this organism. So Bacillus anthracis, dear doctors, it was coming through the cattle, remember? So there were 2 diseases. On the skin, you get that black area called as eschar. That was hide porter's disease. And there was a fatal thing if you inhale the spores called as pneumonia. Which pneumonia? Wool-Sorter's disease. Okay. Next, Bacillus cereus should be known for what? Food poisoning. C for cereus, C for Chinese fast food poisoning. Okay, that's that. Next, coming to the genus called as Clostridium. Okay, now we have the genus by the name of Clostridium. This is what we are going to study now. Let's start from here. If we are coming to this organism, I have already talked about the spores. The spores may have the appearance called as drumstick or tennis racket appearance. So this is the slide as such showing you this. And if you pay attention, they are the bulging spores. Okay. Now starting with the disease tetanus. See, this is the topic we have learned the other day when I was teaching you mechanism of toxins. All right. I very clearly explained to you how does tetanus produce a contraction and how does the botulinum produces relaxation. So one is a contraction, one is a relaxation. How exactly does it do that? That's what we are learning. So here we are, write down the word tetanospasmin. Tetanospasmin. This is the word right here. Okay, tetanospasmin. So technically, what you need to know as such is this, doctors. The word spasm is there in the name. What else do you need? The word tetanus is there in the name. Tetanus means spasm, or spasm is spasm. So 2 times they're shouting at you that it produces contraction. How does it produce contraction? By blocking the release of GABA or glycine. I told you now, what is the normal function of GABA? It relaxes muscles. GABA relaxes muscle. So if you block GABA, the muscles will contract. That is your tetanospasm. Okay. Now clinical features. Clinical features. Please understand. Write it down. It presents with trismus, also called as lockjaw. Okay. It presents with something called as trismus, also called as the lockjaw. Take is that clear enough to all of you? Trismus or the lockjaw. Done. This is the contraction in the jaw. That means the patient complains that I'm unable to open my mouth. That is what? That is trismus. Next, rhiza sardonicus. It is called as devil's grin. Rhiza sardonicus is called as what? It's called as the devil's grin. Okay. So this is what? See, the facial muscles, they contract in such a way that it makes you look evil, and it looks like you're smiling in a very evil way. This is called as rhiza sardonicus. All right. And third and the final, something called as opisthotonus. Opisthotonus. Please see here. In this, the person's head will be touching the ground, and the legs, only the feet will be touching the ground. The remaining body is all suspended into the air. Okay, like this. This is called as what? This is known as the opisthotonus. Is that understood? So head and the feet are touching the ground. Everything else is suspended in the air. That's your opisthotonus. Okay. So what is happening in all the three? It's nothing but a contraction. Do you agree? It's nothing but a contraction. Contraction of the jaw, trismus, contraction of the facial muscles like this, rhiza sardonicus, and opisthotonus. All right. Now we have already learned for the diagnosis. What is the culture media you need to remember? RCM. I told you now these guys want meat pieces. That is what? That is RCM. Robertson's cooked meat broth. Robertson's cooked meat broth. Clear enough? And this is something you all should be striking off, not that important for your exam, so it's okay, you can strike this topic off. Technically in the RCM some color changes happens in the meat pieces, so with that we identify who could it be. Okay, so we memorize it like that. Perfringens pink color hota hai and tetany would be black. Okay, so anyway, that's done. Now coming to Clostridium botulinum. Okay, the next one in the list is what? Clostridium botulinum. And what's the deal? What you need to know here? Clostridium botulinum, doctors, this guy produces a toxin by the name of Botox. It produces this toxin by the name of Botox. All right, and how does Botox work? It blocks the release of ACH. It blocks the release of ACH. And I told you what is the job of ACH? Contraction of muscles. If you block that contraction, what do you get now? Relaxation. Are you understanding? If you block that, you're going to get the relaxation. Okay, so that's that. This is your Botox. So Botox basically relaxes your muscles. Okay, now continuing the story. This Botox, many of you, I'm sure you know it as a drug, right? We use it in the treatment of wrinkles, for example. What are wrinkles? Wrinkles are contractions of your muscles. Wrinkles are produced due to contraction of the muscles. It makes you look old, right? So somebody who gets, wrinkles by age, after a certain age, you start to get wrinkles on your skin. So you take Botox in those areas, the muscles will relax and your skin will lose those wrinkles. You will not get those wrinkles now. So that's the use of Botox these days, which many of you have learned. So technically, what is wrinkle contraction? What is Botox doing? Relaxing it. That's what it is. Okay. But that's all, the medical uses of this toxin. But when this guy comes into your body as a toxin as such during the food poisoning, what can it do? So first of all, what is the food poisoning known as? Botulism, right? This food poisoning is called by the name of what? It's called as botulism. Is that understood what I said? Okay. Now, if you got this idea, botulism, let's now check it out what I'm saying. So first of all, this toxin is coming through canned foods. Of this organism, this food poisoning happens through canned foods, especially the home canned foods. Okay. This usually can come in your body by home canned foods. Next thing, what are the symptoms? What is the presentation? You need to understand one thing. Unlike all the other food poisonings, where you think diarrhea will happen due to, any food poisoning, this one, on contrary, causes constipation. And there's a reason behind this, guys. It's not like, just a fact that you're going to memorize. Think about it, right? When you want to defecate, when you are passing the stool, something is happening in your GIT. What is this movement called as? They're called as peristalsis. When you contract, the feces keep moving forward. When you keep contracting, the feces keep moving forward. Eventually, you will defecate. That means to pass stool, what do you need? Contraction, called as peristalsis. Are you understanding this? If your GIT is relaxed, because what does Botox do? Botox relaxes muscles now. If your GIT is not contracting, if it's relaxed, what do you think will happen? You can't pass stool. And what is that called as? Constipation. Okay, this is going to lead to constipation, right? Your ocular muscles are not held in one place, leading to double vision like this. It's called as diplopia. So all these things, basically, whatever I just, described, they are all due to one common thing. What is one common thing in all of them? Muscles are relaxed. That's the disease. In tetanus, muscles were contracting. In botulism, the muscles are relaxing. And if your diaphragm gets relaxed too much, this leads to respiratory failure. So the patient may die of what? The patient may die of the respiratory failure, doctors. So this is something that you need to keep in mind. Infants can also get this. Infants can get botulism if you feed them raw honey. Okay, you want to feed them honey. See, this is a very common practice in India, right? Feeding honey to the children, babies. These people, if you want to feed them honey in this age group, you can feed them the branded ones, which are pasteurized and stuff. They are not having infection. If you give the raw honey, then the babies may get this infection. All right. So that's about botulism. How do you think the baby also dies? Same reason, respiratory failure. Same reason here. What is that thing? It is nothing but the respiratory failure. So that's all about that guy. Okay. Next, Clostridium perfringens is the next story. Okay, Clostridium perfringens. All right. Previously called as Clostridium welchii. Clostridium perfringens called as Clostridium welchii. Okay. Previous name is welchii. So technically, this guy causes food poisoning. I told you this when we were learning Bacillus cereus. Remember? I told you Bacillus cereus, when it's causing diarrhea, it's copying which guy? Perfringens. Right? So that is that. But it's not important for exam. So don't bother even memorizing it. I don't care. The point is this. Okay. It produces gas gangrene. The most important disease is what? It produces gas gangrene. And this gas gangrene has very high mortality, as you may be knowing. Okay, so hota kya hai? When you go near that gas gangrene area and try to listen, you get this crackling sound called as crepitus. Okay, that crackling sound would be called by the name of crepitus. So let me just show you this. See, this is nothing but your gas gangrene, doctors. You can see the gas bubbles. See, it's a very bad condition. If this happens in somebody, we have to amputate. Basically, we'll have to treat this by amputation only. That too, the chances of survival are very, very less. Okay, so that's this guy. All right, now that was the one where you kind of go nearby and listen. You're going to get that sound that is known as what? Crepitus. Okay, that would be known as the crepitus. Is that clear enough to all of you? Next, once you know that point, there are other agents also, by the way, that can do that. These are the agents who can cause gas gangrene. Next, coming to this point, that is gas gangrene is caused by one toxin which has different names, but usually lecithinase is the name they may ask. Okay, so gas gangrene is due to which toxin? Lecithinase. And to demonstrate the activity of lecithinase, there is one test that I'm pointing at right now. Okay, there is this test that we have for demonstrating the activity of this, and this is called by some name that I want you to learn now. It's called as Naegler's reaction. Naegler's reaction. See, in this there are multiple tests, multiple things we need to identify. So that's why I will show you this. You better put this down. Okay, there is this mnemonic that I'm asking you to learn now. What is this mnemonic? Superstar NTR. Right? Recently there was this hit movie you might have heard about, right? RRR movie. In that the lead actor, there were two actors. One of them is called as NTR. So you're going to remember this word, superstar NTR, because in all the Clostridium, the most fatal is this one, the one that causes gas gangrene, right? The one that is causing gas gangrene. So understand this point. The gas gangrene thing is due to this toxin. Okay, so we have a special test. And remaining, come to this point. What is superstar NTR mnemonic? First one, stormy fermentation in milk media. I'll show you each and every picture, right? First, let's finish. Superstar word for stormy fermentation. Next, the thing I'm pointing at called as Naegler's reaction, the test for the toxin. So Naegler's reaction is a toxin test. Which toxin? Lecithinase. Naegler's reaction. Next, target hemolysis. Target hemolysis. And that target appearance comes due to one factor called as theta factor. Okay, target appearance, superstar NTR, we are in the T right now. The target appearance comes by the theta factor. And last and the final, it is reverse CAM test. Okay, reverse CAM test. A while ago, we learned the regular CAM test. And what did we say the regular CAM test is? Regular CAM test is for the group B strep. Arrow was seen there, yes or no? This time also arrow is seen, but it's from both ends, which gives it a typical appearance of a tie that is worn by the men on tuxedo. What is this appearance described as? Bow tie appearance. You all know now, this is described as a bow, so it's called as bow tie appearance. Okay, reverse CAM test. So now I hope you got the entire mnemonic superstar NTR, starting from first word, stormy fermentation, N for Naegler's reaction, T for target hemolysis, and R for the reverse CAM test. Now let's just have a look at each of these things as such. See, first of all, this is the milk media. You can see that very clearly. In the milk media, it produces this thing called as stormy fermentation. It's producing fermentation in that, which looks like a storm. What happens when you get a storm? The vision is blurred. Are you seeing it's all becoming blurred in here? That is the thing called as stormy fermentation of this guy. Next, the N I already told you, Naegler's reaction is this. This is the test for the toxin. This is Naegler's reaction. T for the target hemolysis. Do you see it's looking like targets in the center? Okay, so this target hemolysis is due to that factor we wrote by the name of what? Theta factor. It's by this thing by the name of what? Theta factor. And last and the final, we just learned something by the name of reverse CAM test. Okay, so this is the thing by the name of what? Reverse CAM test. Okay. Next, continuing the other points. Now you have Clostridium difficile. Okay, next one in the list is Clostridium difficile. So first you write down what are these names written here. Clostridium difficile causes pseudomembranous colitis or antibiotic associated diarrhea. Okay, pseudomembranous colitis or antibiotic associated diarrhea. So that's the word that you got to know, pseudomembranous colitis or antibiotic associated diarrhea. Okay, so I hope you have written this. Now let's look at this concept. Why specifically the name antibiotic associated diarrhea? You could have simply called it that diarrhea, yes or no? So that's the concept I want you to learn. See, this is the concept, doctors. We all have some bacteria inside of our body, normal flora I told you about, remember? So what happens is, if you use very strong antibiotics, the bacteria you're trying to kill will die. That means the infection will die. But at the same time, even your normal flora will die. When you use very strong antibiotics, what could happen is even the normal flora could start dying. So that's where the new organism can come in your body now called as difficile. Okay, difficile now comes inside your body. It gets an easy chance now. Until now, what was the story? Until now, some normal flora were there. They were fighting. They were not allowing others to come inside. But now because this has happened, that means the antibiotic has killed the normal flora. Because the antibiotic has killed the normal flora, now the difficile is getting a chance to come inside. Okay, so if you got this idea, this is your Clostridium difficile what I'm talking about. Hence the name antibiotic associated diarrhea. So the examiner will love to ask you, कौन सी antibiotic से होता है? Which antibiotic causes this problem? Right? Cephalosporins are very commonly used these days. That's why they are the most common antibiotics. They're the most common antibiotics that can do this right now. And very important for exam, more than cephalosporin, although they are more common, in exam they will usually give clindamycin. So with clindamycin, you should definitely know what? Difficile. You give clindamycin, difficile enters your body. Okay, next one. You should remember this. What are you going to do? This disease is also by the toxin, it's not by the bacteria. So better thing is demonstration of the toxin by PCR. Okay, demonstration of toxin by the PCR is a better test that we can do for this time. Okay, now one more very important aspect in this topic is this, guys. Antibiotics are causing this diarrhea. So how to stop it? right? See, a lot of times to stop the diarrhea, to kill the organism, we use antibiotics only. So if antibiotic itself is causing the problem, then how to treat it? That means there are very few antibiotics that can actually kill this agent. So that's where you have to know those guys. Okay, so listen up what I'm saying right now. The drug of choice for treating this condition is called as fidaxomicin. Okay, it is called by the name of what? Fidaxomicin. Fidaxomicin. This is the drug of choice. If this is not available, then oral vancomycin. And this is super important for questions. Make sure you know this. Oral vancomycin. If this is not there, metronidazole. Metronidazole. Okay, so fidaxomicin, oral vancomycin, and then metronidazole. Metronidazole. Is that clear enough to all of you what I said? Okay, so fidaxomicin, oral vancomycin, and then metronidazole. Okay, so this is the treatment as such for this agent right here, what we call by the name of Clostridium difficile. Because it's an antibiotic associated diarrhea, some antibiotics are used to stop that diarrhea. So that's where you have to know them. You have to know them because only few antibiotics are able to stop that diarrhea. Okay, so that's that. Next, we'll be starting with this guy called as Corynebacterium diphtheria. Okay, the next one in the list is Corynebacterium diphtheria. What is the story about this agent now? Corynebacterium diphtheria. See, doctors, this one is also a disease. This one is also a disease which is basically due to the toxin. So it's a toxemia. Okay, so this is also a toxemia. Diphtheria is a toxemia that acts by inhibiting protein synthesis. That acts by inhibiting protein synthesis by blocking one factor we studied yesterday called as elongation factor. Are you able to recall something from the word elongation? Elongation of what? Polypeptide chains. Remember, elongation of polypeptide chains. So if you block that factor, I mean the bacteria blocking that factor, so humans cannot do their protein synthesis easily. So this toxin can do two things. It may damage the heart or the nerves. Okay, so this guy may damage the heart also, or it may damage the nerves also. So I hope this is clear enough as such. Okay, next, coming to the disease called as diphtheria, what are certain things that you need to know as such with this topic? Okay, see, one of the first things you all got to know here is this, my dear doctors. There's something called as Forshell type of diphtheria. It is the one that presents with dirty pseudomembrane. And I'm positive you definitely must have seen this picture for sure, right? This is the dirty pseudomembrane that is present with the diphtheria. Okay, now going back to this, what you got to know, apart from this, another very important feature is this appearance, what you call by the name of bull's neck appearance. Okay, this is described as the bull's neck appearance. The dirty pseudomembrane, bull's neck appearance, these are the two things. And technically, I just told you this guy damages heart, because of which you're getting myocarditis, right? You're getting myocarditis. Myo means muscle. So technically, if the muscles of the heart are damaged, you cannot circulate the blood. What is the job of heart? Pump blood, circulate blood. So that's why, what is the cause of death in this person? Circulatory failure. Please make sure you know this. Diphtheria kills you by what? By circulatory failure. Okay, so that's something that you also be familiar with. Okay, now coming to the diagnostic aspects. Okay, diagnosis. First of all, know one thing here. If you do gram stain, or let's say if you take a special stain called as Albert stain, either ways, the point is it will be showing something by the name of Chinese letter pattern, right? This guy will be showing what pattern? Chinese letter pattern. And what you need to know is the gram stain obviously will show it in purple color because it's a gram positive agent. And Albert stain is a special stain. Yesterday I did not teach you that. I just showed you that this Chinese appearance we will talk in diphtheria. Now I'm going to talk about this. Okay, so something which I may not have covered before, I told you respectively in each topic we'll talk. So here's where this is that Albert stain, and these ones, the way they arrange themselves, the rod shaped organisms, the way they arrange themselves, like you can see here, this is described as the Chinese letter patterns. Like, you know, this random appearances that are appearing, these are called as what? These are called as the Chinese letter pattern. Is that clear to all of you? So this is it. Now, if you examine this picture clearly, you also get to see these round things now. What are these round things that you're seeing? These are some granules. So in Albert stain, you also get to see something called as granules. Okay, so what is special with the granules? They are energy granules, which are given a name called as metachromatic granules. They are called by the name of metachromatic granules, also called by some other names. Okay, so point is, because diphtheria is a fatal disease, usually when there is something fatal, some special tests would be always available. Like Superstar NTR, we talked about so many things, right? So same way, that was a fatal disease, gas gangrene. Here also, these are the granules you got to know. Next, Loeffler's serum slope. Now, what is the story behind this? LSS stands for what? Loeffler's serum slope. So this is basically an enriched media we talked about. Today only we talk culture media. I've explained to you. One is dal chawal, simple media, and to the simple media, when you add some things like blood, serum, it is called as enriched media. So this is the Loeffler serum slope. Serum has been added. Everybody loves this if you add something extra. Okay. Now here we are. This is the selective media. So what is the difference? Loeffler serum slope is enriched media. It's not only for diphtheria. It's not only for the diphtheria. Whereas the selective media is PTA, which is only for diphtheria. Okay, PTA. What is PTA? Potassium tellurite agar. Potassium tellurite agar. That's your selective media. That is PTA. Potassium tellurite agar. I wanted to make sure you write this clearly. What is the advantage, disadvantage? You will come to know right here. Okay, within this area, you will come to know what is the advantage and disadvantage. So guys, LSS is what? Loeffler serum slope. It's not selective for anybody, right? So that is a faster growth. That's the advantage. On LSS, the growth of diphtheria comes within how much time? 8 hours. Of course, they like extra things, so they will grow faster. Selective media, it takes time to isolate them, but it's the best one. That means this media, if it grows, it has to be diphtheria. It cannot be anybody else. So that is the word called as selective media. PTA, potassium tellurite agar. And in yesterday's topics, I very clearly explained to you that usually when you do the culture, how long does it take for the report to come out? I said 2 to 3 days. Do you remember that point? 2 to 3 days. That's what is the deal here. Nothing special, right? So LSS is 8 hours, PTA is 48 hours. One is 8, another is 48. So that's your PTA, potassium tellurite agar. And how long are they taking? 2 days. Done. So that's about this guy right here. Okay, so this points you may strike out, guys. It's usually asked in PG, you don't need to know. Now, toxin demonstration, how is it done? See, a while ago when we learned perfringens, somebody called by the name of perfringens, what did I tell you back then? I told you that perfringens had something for its toxin called as Nagler's reaction. Remember superstar NTR mein, and for the Nagler's reaction, whereas this guy, you have this test by the name of ELEX test. Okay, so here's where you have the ELEX test. Yeah. Now you have Schick's test. We don't do it anymore, but just write that name. It was done when the vaccines of diphtheria were very limited. Only people who they thought are more prone to getting diphtheria were, given the vaccine. So for that, they used to check your immunity by doing one test called as Schick's test. Now the diphtheria vaccine is abundantly available. We don't do that test. Imagine Corona beginning. Don't you remember those were the days when the waiting time for the second dose was so long because the vaccines were limited that time. We wanted each people to get their first dose before the other people get their second dose. Yes or no? That's why the second dose, the time was more that time. So that's the thing I'm just telling you here. Schick's test was a test like that, which we used to do before, not anymore. But agar, just for a reference as a doctor, not everything is important for exam in the life, right? There are things that you got to know when two people talk. When doctors are talking to each other, you have to know some things that happen. It's not something very, very rare. Schick's test was a popular test. That's why you have to get this idea. Okay? Now, if you have finished this, coming to this very important test. So for this, first I would want all of you to note this thing down, what I'm showing you. Okay, so please note this thing down, what I have written there. ELEX test, change the spelling to ELEX. Then what are you looking for? It's written there. Look at the middle strip. In the middle strip, if you see the X, okay. So I hope you guys are done. I said there is a test for the toxin called as the ELEX test. Change the spelling to ELEX because that's what you're looking for in the test, X. If you see the X, then that's toxin producing. And in this picture that I have given you, I want you to please mark it. I want you to mark this here. Upper and the lower strip is the control and the middle strip is from the patient. Middle strip is coming from the patient. So upper and the middle is the control and the middle strip is the patient one. And if you see in the sample, very clearly you will be able to appreciate there is a shape of X forming around the patient. That means this is a toxin producing strain, and that's a bad news because toxin is the one causing the disease called as diphtheria. Did you just understand what I explained over there? Right? This is the toxin producing strain. This is the toxin producing strain. Okay, so that's all about it. If you did not see the X here, assume if it looked like this. If I remove the control word so that you don't get confused, assume this is the middle strip. Are you seeing any X around here like this? No. If you're not seeing the X, that means it's not a toxin producing strain. But if you see the X like this, that means it is a toxin producing strain. So that's as simple as that. That's your ELEX test. Is that clear enough to all of you? So that's all about this organism called as diphtheria. Okay. Now here's the time where we'll take our break, guys. We'll start by 1:40 now. Okay. So 1:40 we'll be starting once again. So see you all then.